Good morning, everyone. It is my pleasure to welcome all of you for today's National Conference on Recent Trends in Applied Aspects of Biological Sciences 2021. I welcome all the eminent speakers and guests from different walks of life who have come here to share their knowledge and vast experience with the student community.
For prayer is nothing else than being on terms of friendship with God. Now I call upon Kritika for the prayer song.
इतनी शक्ति हमें दे न दाता, मन का विश्वास कमजोर होना। इतनी शक्ति हमें दे न दाता, मन का विश्वास कमजोर होना। हम चले ने एक रास्ते पे हमसे भूल कर भी कोई भूल होना। इतनी शक्ति हमें दे न दाता, मन का विश्वास कमजोर होना। मन का विश्वास कमजोर होना। Now, I request our HOD, Mrs. S. Ambika, Head in Charge, Department of Biotechnology, for the welcome address. Please, ma'am.
Thank you. Good morning, everyone. Most respected Secretary, sir, our beloved Principal, respected Vice Principal, eminent speakers, guests, and my dear students and colleagues, good morning. It's my pleasure to welcome all participants today for the 2 days conference on recent trends in applied aspects of biosciences. The theme of the conference is to disseminate knowledge in the recent trends in biosciences. The Department of Biotechnology, Government of India has planned to execute National Biotechnology Development Strategy 21-25 with a theme of knowledge and innovation-driven bioeconomy. India will be in top 5 countries globally as a biomanufacturing hub by the year 2025 to achieve a growth of 150 billion US dollars. They have planned to achieve this with 4 major verticals, likely capacity building, both human resource and infrastructure, strengthening and nurturing strong basic research, innovation-driven ecosystem, product development and commercialization, basic and translational research for new knowledge. So currently, we are in the 12th position as a biotech destination, and we rank 3rd in Asia. So in the past few months, the COVID-19 pandemic has impacted every sector of our economy. However, despite the many challenges that have emerged due to the pandemic, the key growth have emerged intact. So this situation has presented some significant challenges to the scientific community. And at the same time, this has offered opportunities for the pursuit of new scientific activities, particularly in biotech. Finally, now we have begun to reap the benefits of various scientific and medical advancements made in the past. Now let's all join in the path which leads the way forward to achieve success in all other areas. So I congratulate the organizers for arranging such a wonderful conference and giving an opportunity for the students to know the current status during this pandemic period and growth of biotech sector and knowing the importance of the biotechnology industry. So, once again, I welcome you all, and I hope you will all utilize the most of the conference. Congratulations again, and I wish all the participants all the best. Thank you.
Thank you, ma'am. Now I call upon Ms. Satyabhama, Assistant Professor, Department of Biotechnology, to introduce the first speaker of today's session. Ma'am, please do the honors.
Good morning, dear delegates and participants. So, I welcome you all to this wonderful session of the National Conference. So, it's my honor to introduce the Chief Guest of the hour, Dr. Mukesh Tandave. And we have two Chief Guests as of now, Dr. Mukesh Tandave and Mr. Ganesh. So, to start with, Dr. Mukesh Tandave, he is an Associate Professor in the Department of Pharmacology at Delhi Pharmaceutical Sciences and Research University, New Delhi. Dr. Nandave earned his PhD in pharmacology from AIMS, Delhi, and received his postdoctoral training from the Division of Cardiothoracic Surgery, the Ohio State University Medical Center, USA. So, more than 12 years, Dr. Nandave has been investigating the role of nutraceuticals, herbamineral formulations, plant extracts, and constituents for myocardial ischemia and reperfusion injury, diabetes, obesity, pain management. So he has published more than 65 papers in peer-reviewed national and international journals. So his citation are now at the range of 808 with an H index of 18. So his lab received more than 1.5 crore total funding from the government of India as well as industry, including Palmer's, Dabur, Charak, Madhobag, Sandhu, etc. So Dr. Nandave has received numerous awards, including G. Achari Gold Medal by Indian Pharmacological Society, Association of Pharmaceutical Teachers of India, Young Pharmacy Teacher of the Year Award, the Indus Foundation's Award for Research Excellence, Early Investigator Award by International Society for Heart Research, Professor Dugirala Visveshwaram and Professor Srimantula Satyanarayana Prize, Best Research Output of the Year for the year 2014-15 and 2013-14 and for 2012-13. And he is a member of the International Society for Heart Research, International Academy of Cardiovascular Sciences, the Indian Pharmacological Society, the Indian Pharmaceutical Association, Association of Physiologists and Pharmacologists of India, and Association of Pharmaceutical Teachers of India, and the Society for Ethnopharmacology. So I welcome Dr. Mukesh Nandave, and I also thank him for promptly accepting our invitation and to enlighten the students with the emerging trends in pharmacological sciences. We welcome you, Dr. Mukesh Nandave.
Yeah, good morning to one and all. And it's really a great honor to be very dynamic audience and the organizing committee, uh, principal of the institute, and faculty and students. Uh, I'm really honored to be joining you in such a dynamic research and a scientific program. Uh, Also thankful to Dr. Satyabama giving me this opportunity to talk to your students and faculty members. So today I'm going to talk about how biotechnology changed the approach of doing drug discovery and development. I hope I'm audible and clear.
Yes, sir, you're audible and clear. It's
Okay.
perfect.
Yeah. So, so today I'm going to talk about the various biotechnological approaches that help to speed up, that help to what you call accurate the drug discovery and development process. And we all know that it's a very long, what you call tedious and costly process. So let me share the presentation. Uh, Okay. Is my screen visible?
Yes, sir, it's visible.
Okay. So, history of drug discovery is not new because in the past also many what you call pioneers in pharmacology, then pioneers in drug discovery and development, or before than that, in many centuries, uh, what you call the Ayurveda, Chinese medicine, and different systems of traditional systems of medicines contributed. Okay, in fact, thousands of years back, uh, human beings started with the use of plants, then marine drugs or drugs from the animals. Okay, so basically, what they did, they used they followed the surrounding, and someone rightly said, when you want to find the solution for your problems, you have to look at your surrounding, your nature, your ecosystem. Okay, so in a similar manner, we found the drugs from our what you call plants, then animals, marine sources. Okay, and if you see this, what you call as early as the 5th century, the Greek physician Hippocrates, okay, wrote about the ability of powdered extracts from the plants, and they were used for the treatment of diseases. And then, if you see, the most important or well discussed example is of a willow bark. That bark of willow was used to cure or uh, alleviate the aches and symptoms of fever and other infections. But then uh, it was not standardized, like how much extract of the bark. Okay, but then later we found, with the help of extraction methods and various apparatus and tools and technology, that complete willow bark is not required. In fact, the salicylic acid present in that is more important. Okay, but then when they isolated the salicylic acid, they found that it is effective, but it is equally unsafe also. Okay, and then they synthesized the acetyl salicylic acid, what we know today aspirin. Okay, and how aspirin acts, that is discovered by again a scientist, and he got the Nobel Prize. Okay, he proved that aspirin acts through the what you call inhibition of cyclooxygenase pathway, COX1 and COX2 pathway, or arachidonic acid pathway. So this is how we develop. Then similar with the morphine, okay. Similar with the sulfonamides, okay. So so this is about the history of the drug discovery. If we talk, the traditional Chinese system and Ayurveda both several thousand-year olds, and in that systems, we have used the plants, animal sources, and marine sources for getting the drugs and treating the disease conditions. And aspirin remains the most discussed or famous example. Okay, even the inoculation is at least 2,000-year old. But what is difference between the drug discovery in the 21st century and then in the past? That in the past we were not that successful to get the efficacious and safe drug. But yes, because of the stringent regulations, because of the more and more biotechnological omics techniques and tools, okay, we can precisely say that what is the mechanism responsible for the particular disease and how it can be targeted by targeting the receptor, enzyme, or whatever the druggable target. Okay. So in the past, we were uh, having more ineffective drugs, but in current time, we have precise, selective, specific drugs which are better in terms of safety as well as efficacy. Okay. Now, these are the few more recent efforts, like in 1796, Jenner found the first vaccine where he demonstrated that cowpox prevents the smallpox. Okay, and then exactly 1 century later, Pasteur made the vaccines against the anthrax and rabies. And then we all know that Paul Ehrlich, who is considered as the father of chemotherapy, discovered that industrial dyes even can be used as antimicrobial therapy. So sulfonamides were developed for antimicrobial purpose in 1930. In fact, Sir Domark got the Nobel Prize to discover that how these sulfonamides acts and can be used as antimicrobial chemotherapy. So, in the second half of the 20th century, we started using the modern chemical techniques to create the more and more safer and efficacious drugs. So, what we have very rational approach of drug discovery that is basically a process finding the new chemical, maybe a new biomolecule, or maybe a new enzyme, vaccine, or whatever. Okay, but basically it's a process of finding the new medication based on the knowledge of a biological target. Okay, we don't use the random approach of drug discovery. Okay, we use the rational drug discovery. Like this is the most incident drug, disease problem. So these are the various targets responsible means involved in the disease pathology and etiology. Now, which is the what we call most crucial target, which inhibition or activation will lead to the major impact on the disease outcome. Okay, that we find out, and once we know what is the target to be targeted, then we try to find out the organic small molecules which can activate or inhibit the function of that target. So obviously, most of the time targets are proteins, like enzymes are proteins, hormones are proteins, receptors are proteins. Okay, so these proteins are either activated or inhibited by your synthesized compounds. And it also involves the design of molecules that are complementary in the shape and charge to the biomolecular target. Okay. So obviously the key and lock and key concept always work there. Uh, for any receptor to be activated or to be inhibited, you have to find out what is the binding site and what is the conformation of that binding site. And accordingly, complementary molecule has to be designed, synthesized, and tested its interaction against that established target. Now, if you see, these are the timelines. Most of you may be little knowing that we have the three main stages of drug discovery. That is stage 1, stage 2, and stage 3. So in stage 1, it's all about the drug discovery. Means you find out the target, you validate the target, you taste it whether it is a druggable or not. Okay, and then once you establish that target, we carry out the chemistry and taste the synthesized compounds or molecules against the validated drug target. Okay, and you carry out the efficacy and safety studies that may be in vitro, that may be in vivo, that may be in silico, that may be ex vivo. So in vitro means in cell lines, in what we call ex vivo means in the tissue organs, in vivo means in a small to large size animals, and what we call in silico means that is on a computer with the help of bioinformatics. Okay, and various computer programmings. Okay, and then once we found that the drug is efficacious and safe in animals, in cell culture, in silico setting, in what we call organs and tissues, then we can get the approval from the regulatory agency that yes, our compound or the new molecule is efficacious and safer and can be tested further in humans. Okay, and that is stage 3, what we call clinical development. So drug discovery mainly indicates that all the studies in animal cell lines and computer programs, okay, where development is means the develop is testing into the humans, okay. So this, the funnel itself indicates that the success rate or the what you call difficult level increases as we progress from stage 1 to stage 3. So what I mean, we start with the 10,000 compounds and we end up with the one successful drug that is used in a what you call patients or humans. Okay. Now, out of maybe if there are 100 what you call compounds or the drugs that are FDA approved, maybe only 30% of the drugs gives you return on investment. Okay, so when we start with the 10,000 compounds, we can get the one successful drug to be used clinically. And out of 100 clinically approved drugs, only 30 drugs can give the return on investment. Means whatever the amount spent, whatever the time spent on developing the one successful drug, maybe only 30% of the drugs can give the return on investment to the what you call sponsor company or the innovator company. Okay. And if you see the timelines, more than 10 years required to develop a one successful drug. Okay. And so this is what very difficult process, very lengthy process, and most importantly, very costly affair. So it's not the cup of tea of every company. And that's why not many what we call Indian pharma companies are playing in this drug discovery and development area. In fact, we go through a different approach like the generic drugs, okay, or maybe the repurposing purpose approach. Now, if you see the economics, okay, economics, if you see the number of drugs that are what you call approved, okay, are declining, okay. In fact, number of drugs per billion US dollar R&D spending is what you call decreasing. Why? Because after the thalidomide case, FDA tightened, or in fact, almost all the regulatory agencies tightened their rules and regulations, and that's why not all the drugs not all the drugs can pass all the stages of the drug discovery and development and cannot reach to the market. Okay, so this is what. So in 1950, maybe more than what you call 50 drugs would be entering into the market. But if you see in 2010, like in 2021, the scenario may be very different. Okay, so this is what the economics. So the drug discovery and development process becoming costly and costly year by year. Now, if we see what is the cost consumed at every stage of the drug discovery. So if we talk, we have the discovery of the basic research phase, then preclinical testing, clinical trials, and under the clinical trials, you have the different phases or stages. Okay, so if we see 10 plus 14, almost 24% of the expenditure goes into the discovery and basic research of a drug. Then If we talk about the preclinical testing in animals and cell culture or formulation of the dosage form, almost what we call 12% of the expenditure goes in this. And the most costly affair, the stage, is the clinical trials. Okay, why? Because it's the first time in humans, and as we progress from the stage, phase 1 to phase 3, we have to increase the number of patients or the volunteers that taking part into the study. In a short, we have to study the drug in a large number of population for establishing its efficacy and safety. And that's why the clinical trial is the most what we call costly stage of the what we call drug discovery and development process. Now, if I say it's a very risky and very tedious. Why? Because from any chemical compound to get a successful drug candidate, you have that drug candidate has to meet these requirements. Means it has to be very sufficient efficacy. That compound should show acceptable safety. It should be metabolically stable, chemically stable. Its absorption should be in a such manner that acceptable bioavailability is achieved by that compound. And solubility is also important criteria. So fulfilling all the requirements by any compound is really difficult. Okay, and that's why the progression of the hit to any lead becomes very difficult because of this number of parameters. Okay. Now, because of this tedious process, many drugs fail during the process, and in fact, many times even after the drug approval. So, major reason of drug failure in a drug discovery and development process is efficacy. The second reason is safety, and then strategic, then commercial. So what I mean by commercial, commercial means yes, you have the drug B already in the market by a competitor, and you are in the process of developing the drug A. Now, during the drug development, discovery and development process, if you found that your drug A is not that much effective compared to the already available drug, then there will be no future of that drug. Okay, why patient or the doctors will use the less effective drug compared to the already more efficacious drug? Okay, and that's why industry or the innovator company don't want to take a risk of putting more time, more money into a less efficacious drug. Okay, so that's a commercial that they don't see a bright future on return on investment of that drug under development, and that is why they halt the process of for that molecule. Okay, so that is what the commercial. Now, this is what the complete discovery and development process, where we start with the target validation, and against the validated target, we study, we screen the compounds, chemical compounds synthesized in a chemistry lab, or maybe they are isolated from the plant, or maybe biologically produced, and then we find out the heat. And when we study the heat more and more in the detail, then we get the lead. That is what the lead generation. Okay, and then this in the heat identification and lead generation, you have a major role of computational chemistry and medicinal chemistry. Okay, and then those compounds successfully show a better binding to the validated target. They have the chemical and metabolic stability. They have the absorption. They can be absorbed. They can be what we call solubilized. Then only we take that hit molecule to the lead stage and what we call lead candidate. Okay, So lead optimization, again, we study the pharmacokinetic and pharmacodynamic of the drug, that lead molecule, and then we take that to the preclinical development into the small to moderately or large size animals. Okay. Here, for optimizing the lead, we have to what you call carry out the DMPK and bioanalysis. Structural and analytical sciences also plays an important role. And then pharmacology and safety pharmacology is also studied of this lead molecule. Okay. Then simultaneously from this stage, we work on an API development, that is active pharmaceutical ingredient development and manufacturing. Then pharmaceutical development and manufacturing, toxicology, pathology, okay, and clinical sciences. So all this process goes hand in hand to get a successful drug in the market. Now, these are the various approaches, okay, used in a drug discovery and development before the 20th century, during the 20th century, and in the 21st century. So in 20th, before 20th century, we used to study the drug based on the bioassays, pharmacokinetic and pharmacodynamic studies, and ethnopharmacological studies. Okay, but later we became more vigilant. In fact, regulatory agencies developed stringent regulations, and in 20th century, addition to this, we have to establish the safety pharmacology of that drug. Okay, and then reverse pharmacology means the drug which is already used clinically by maybe other systems of medicine, that drug can be taken from the clinic to the lab and establish that it is really have a scientific basis and can be used scientifically and clinically into the patients. Okay, and then you have the in the 21st century we have the network-based approaches, omic science, personalized medicine, system pharmacology, and artificial intelligence. Okay, so this is what the change of approach from before 20th to the 21st century for drug discovery and development. Now, how this biotechnology plays a very important role, or how this biotechnology changed the approach of drug discovery and development. Okay, so if you see, the biotechnology produced more than 200 new therapies and vaccines, including the products to treat cancer, diabetes, HIV, and autoimmune disorders. So if we see in cancer, the monoclonal antibodies, okay, change the treatment radically, okay. And in fact, in the diabetes, okay, earlier we used to depend on an insulin from the animal sources. But what was the problem of insulin from the animal sources? The exact similarity of human insulin and the insulin from the animal source was not there. Okay, and because of that, humans used to show the hypersensitivity reaction or allergic reactions to the insulin produced from the animals. The another thing, we has to more and more depend on animals for taking out the insulin. Okay, but now because of the recombinant DNA technology, we can produce the exactly similar insulin what we need for humans, and we don't have to depend anymore on animals. And another most important thing, that because of the different biotechnological approaches, we don't have only the human insulin, we have insulin analogs as well. Okay, we have the different combinations of insulin. Means insulin analogs means earlier we started with the human insulin, and then we have the Lispro, then Lantus, and other types of insulins. Okay, like what is the difference between this human insulin and then insulin analogs? So if any patient of diabetic patient is using the human insulin, that person has to take the dose before the meal, at least 15 to 20 minutes. Okay, because it is this human insulin shows the action very slowly. Okay, and that's why to cover the blood glucose, you have to the patient has to take in advance the dose of the insulin or injection of the insulin. But with the insulin analogs, you can take the shot, insulin shot, with the first bite of your meal. Okay, because they are very rapidly acting. Okay, very rapidly acting. That's why they take a quick control of blood sugar. Okay, so that is how the biotechnology changed the insulin production and change the different types of insulin. Okay, more than 400 biotech drug products and vaccines currently in a clinical trials targeting more than 200 diseases, including various cancers, Alzheimer's disease, heart disease, diabetes, multiple sclerosis, AIDS, and arthritis. And average approval of 10 to 15 products a year that indicates that pharmaceutical biotechnology is very or highly productive sector. Okay, and biotechnological companies are said to be more entrepreneurial, nimble, and very fast. So if you see, in case of the vaccine, COVID-19 vaccine, okay, so many vaccine candidates are already entered into the market, and you can see their efficacy as well. Okay, and how they are effective in what you call preventing the COVID-19 infection, okay, or corona. So that is how the importance of biotechnology in drug discovery. Now, if we talk about the Indian biotech market, so India ranks 12th in the world and third in Asia Pacific region. Almost 3.7 billion US dollars to be spent on biotechnology from 2012 to 17, and we are the number one producer of hepatitis B vaccine from the recombinant technology. And by 2025, our Indian biotech industry will be more than 100 billion US dollar. Okay, so we have a very bright future. Now, these are the just a few examples of top five most expensive medicines. And in fact, maybe last week only, many of you read about the most what you call most costly drug. Okay, that is for very rare disease reported into children, and the single dose goes into the crores of rupees. Okay, now in drug discovery development process, biotechnology has two main applications: that development of the traditional small molecule drugs, okay, where it biotechnology allows the researcher to focus on a drug discovery on a single molecular target. Okay, so earlier it was a random approach, means you synthesize the compound, you synthesize the compounds in the chemistry lab and screen them randomly on available drug targets, okay, in a particular disease. But now, because of the human genome projects, we exactly know what is the what you call gene and what is its function and how it is adversely changed, and that leads to the disease. So we know the root cause, and that's why we can precisely develop the drug against the single molecular target. Now, techniques include molecular biology, biochemical, and biophysical methods that helps you to establish a single molecular target. Now, this narrow down approach is better. Why? Because you have high chances of getting successful. Okay, because we know that this target is really crucial for treatment of that particular disease. Okay, And that is why the biotechnology is used in target identification, target validation, and target protein expression. Okay. In fact, generation of the drug screening assay is also possible in current time because of the biotechnology. Okay. Now, the second application is development of protein therapeutics, or you can say therapeutic proteins. Therapeutic proteins, insulin is the most important example. Then monoclonal antibodies, again, they are the therapeutic proteins. Okay. So compared to the synthetic small molecules, biotechnology can help you to develop therapeutic proteins or protein drugs or protein-based drugs used in the treatment of various diseases. Okay, so traditional small molecule approach plus recombinant technology in the synthesis or expression of the protein therapeutics like antibodies. Okay, so these are the two areas or two steps where the biotechnology contributes a lot and gives you very better results. Now, various technologies for target validation. So we know, uh, as we know that particular gene is responsible for the particular disease, then we can exactly produce the similar disease in animals by knocking out or knocking off that particular gene. Now, when you knock in, we call the knock in mouse, and when you remove that particular gene, that is knockout mice. So, impact of gene deficiency on induction of the disease in knockout mice gives important clues on the causative role of that target. Okay, and conditional knockout means disease induction in the adult animal general switch with the gene in the on state, or the gene is turned off by what you call small interfering RNA. These are the technologies used in production of the transgenic animals. Okay, and that helps you to study your drugs that may be of a small molecule or that may be of a proteinous in nature. Okay, so that is what the technologies for target validation. Now, what is the role of biotechnology in a drug targeting? So targeting using specific vector molecules. Okay, and vector molecules have the affinity towards the ligand characteristic for the target tissue. For example, antibodies, peptides, lectins, and hormones. So, if we see, this is the again the drug delivery. So, immunotoxin, like A and B, they are combined, then splitting. They both are separated, and then the A will fuse with the antibody, and that is what the immunotoxin. Okay. Now the active this A is the active subunit and B is the cell binding subunit. Now this can lead to the specific value. Then you know the catalytic antibodies. So catalytic antibodies obviously used in a treatment of cancer. So compared to the normal cells, cancer cells express the particular antigens. And that against that particular antigen, if you develop the antibody, that is what the catalytic antibodies. Now, what is special between the catalytic antibody and the monoclonal antibody? So in catalytic antibody, we have the antibody, and then we have the enzyme also, and we have the what we call drug reservoir also. So as the antibody will specifically bind to the antigen which is present on the cancer cell. Okay, so delivery of the anti-cancer drug can be also specifically targeted to the cancer cell only. Means the catalytic antibody which is linked with the drug reservoir that contains the anti-cancer drug, okay, will specifically bind to the antigen. The enzyme will activate, and then the drug will be released to that cell where that antibody bound it. Okay, That is what the catalytic antibody, a specific selective approach of delivering the anti-cancer drug. Okay. Now this is what the another approach for what you call delivering the drug. So oligonucleotides and then cell-penetrating peptides can be incubated, and based on their molar to charge ratio, they can form the complex, and then they can, through the cell assembly, can form the peptide-based nanoparticles, which can be what you call transported across through the membrane, and again they what you call dissociate, and then the specific oligonucleotide nucleotides are delivered to the site of action where it is really required. So this is the very specific and effective approach of delivering the oligonucleotides with the help of cell-penetrating peptides. Then there are the other pathways for delivering the drug, like direct translocation across the membrane, then clathrin-mediated endocytosis, okay, and then Kevlo-mediated endocytosis. Then you have the macropinocytosis, then pore formation model, then carpet model, and then inverted micelle model. So through these different approaches, we can deliver your what you call cell-penetrating peptides, okay, to different cell organelles. Okay, it depends upon what is the drug and the amount you have to deliver into the cell. Now, the another application of the biotechnology, that is high throughput screening. Okay, so most of you already know that it's an automated machinery or setup that is used in screening of large number of molecules, maybe thousands to millions of molecules within a 24 hours. Okay, so basically in high throughput screening, you use the multiple well plates that may be 96, that may be more than 1500 wells in a one plate, and you with the robotic arms, automatically the compound is picked up and then what you call added into the number of wells, and after reaction of the molecule with the target, that is target acid set up into that plate, a multiple well plate, will show the maybe absorbance, fluorescence, or luminescence, which will be detected by again a detector, and automatically the data will be processed. Okay, so if we see this high throughput screening really increase the outcome in the early stage of the drug discovery. Okay, so if we see usually 10 raised to 3 to 10 raised to 6 small molecule compounds can be what you call screened in a day, and that compounds can be extracts, can be chemical compounds, can be oligonucleotides, can be antibiotics, and this this STS can really speed up the screening process. Okay, now when we carry out the primary screening, then we will get the few molecules which are really compatible to the your validated target and can be taken further to more number of assays and maybe to the ex vivo studies, to the in vivo studies. Okay, so you have the qualitative high throughput screening and you have the quantitative high throughput screening. In qualitative, the assay, STS assay will let you know that whether compound binds to that target or not, and upon binding, whether it activates or inhibits the target or not. That is what the qualitative. And quantitative, how much compound or how much, to what extent that the target is activated or inhibited, and how much compound is required for optimal activation or inhibition. So that is what the quantitative. So in high throughput screening, cell-based assay or in vitro biochemical assays can be done. And in a cell-based assays, there are three main broad categories. That is second messenger assay, reporter gene assay, and cell proliferation assay. Okay, so this technology again help to screen more and more number of compounds in a short span of time. So Ann et al. Studied the G protein coupled receptor activation using beta galactoside enzyme complementation technology. And by means of flow cytometry, Waller et al. Used the technique for studying the GPCR, G protein coupled receptors assembly, pharmacology, and screening. So this is what the contribution of again the biotechnology, especially high throughput screening in screening of a large number of compounds in a short time. Now there are different technologies for detection. Now detection can be based on the FRET, that is fluorescence resonance energy transfer, or maybe fluorescence polarization, time resolved fluorescence, fluorescence lifetime measurement, fluorescence correlation spectroscopy, fluorescence photo bleaching, recovery, and molecular beacons. So this is the typical platform or the infrastructure of the STS, where from one end the compounds, the plates, and the what you call assays are set up, and then robotic arms actually add the molecule to be tested or the drugs to be tested. Not drugs, because we cannot call drug at that early stage. We can only call the chemical compounds. Okay, so that is what is studied into the ASTS, high throughput screening technology. Now, applications of fluorescent techniques is to identify the protein interaction, monitor intercellular signaling activities in real time, then survey the various bioactive molecules, which has a role in different pathways. In that pathways plays important role in health and disease. Then even dissociation constant of any particular compound can be identified by fluorescence techniques. Then competitive binding assay. Okay, so competitive and non-competitive. So competitive means presence of another molecule will affect the binding of this the another molecule. Means there are compound A and B. So compound B is already bounded to the what you call target, but when compound A is added, then because of A is having the more affinity to the target, A will displace the B and bind to the target. And this is how the competitive binding assay also can be studied by fluorescence techniques. Now, another application of biotechnology in drug discovery is 3D cultures. Okay, so I already told you what are the various reasons for failure of the drug molecules. Now, at present in STS, high throughput screening, we use the 2D cell culture, what you call method. Okay, and 2D cell culture is not the exact representation of the physiological situation in humans. Okay, see, whatever the situation of the environment present in our body is not exactly produced by using the 2D cell cultures. And that's why if you study the molecules or the compounds into 2D cell culture, and even though they successfully pass that screening, these compounds can fail in advanced stages of the drug discovery and development because they are not exact representation of our physiological situation. And that's why people are thinking that 3D cultures, cell cultures can be the solution for increasing the success rate of the compounds. Okay, so 3D cell cultures provide more accurate environment where cell resides in the tissue. Okay, and 3D cell cultures grow cells into 3D aggregates or spheroids using scaffold matrix or scaffold free matrix. And there are most commonly used scaffold used in 3D cell cultures. BD matricial basement membrane matrix from the BD sciences, then Cultrex, that is the basement membrane extract from the BME Trivision, and then hyaluronic acid are commonly used biologically derived matrices. And apart from that, polyethylene glycol, polyvinyl alcohol, polylactide co-glycolide, and polycaprolactone, they are the most commonly used materials to form synthetic scaffolds. Okay, so the one more example that the 3D cell cultures can be can replace means can reduce the use of animals or or organs or tissues excised from the animals. Okay, so that is what the advantage. Now, 3D cell cultures as biosensors, they are a simple, fast, and cost-effective alternative to large-scale and cost-intensive animals testing. That is what I already said. Cells grown in a 3D culture can be incorporated into biosensor either by direct attachment onto a biotic or a biotic substrate surface, or by indirect attachment via entrapment in biocompatible biopolymer. Now, substrate selection depends on both the cell line and the sensor application, and most biosensors use silicon, glass, or plastic surfaces. And the most commonly used transduction technique for 3D cell-based biosensors is electrical or electrochemical biosensor. So this is another application of 3D cultures used as a biosensors in drug discovery and development. Now, bioinformatics, another discipline, sub-discipline of the biotechnology, where PyMol and RASMol, these are the programs, okay, to study the structural orientation or the structure of the molecules. Okay, in fact, which molecule will fit best to the target can also be predicted by use of PyMol and RASMol programs. Then the gene logic, that's another what we call bioinformatics based program. What we call for understanding the underlying mechanisms of diseases, then discovery and prioritization of the gene targets and biomarkers. So gene logic basically, although we know that Human Genome Project already help us to know what are the genes and what is their importance and what is their function. Okay. Then Clindex is an e-clinical trial. So rather than managing the clinical trial from the start to end manually, you can use the Clindex, that's again the program, e-tool, which will help you to speed up, smartly manage the clinical trials through the Clindex program. Okay, so there are so many programs used in clinical trials to manage the data, to study the data, to extract the data, to process the data in a different manner for different objectives. Okay, so that is what the role of bioinformatics in what we call a drug discovery, particularly Clindex, is at a clinical development stage. Now, protein engineering in drug discovery. Uh, Protein engineering improves the pharmacodynamic profile to obtain the drug that acts faster or slower. Okay. See, imagine you have an analgesic drug used in treatment of relieving the pain or the headache. Okay, so if you know that your drug is going to reach into the systemic circulation maybe after 20 minutes, so you should expect the effect, pain relieving effect or headache relieving effect after 20 minutes. Why? Because only presence of drug will ensure that it will be effective. Okay, and that is why that is the what the importance. Okay, now if you can study the pharmacokinetic and pharmacodynamic of the drug by means of protein engineering, you can precisely tell that at what time you should take the drug and at what time you should expect the effect of the drug. Okay. Development of pharmacological half-life and development of a controlled release kinetics can be possible by protein engineering. Then alteration of the receptor binding specificity. See, receptor binding specificity is really important. Okay, why? Because any drug is efficacious and more safe is because of again its specificity. Means specificity is one of the reasons. There are many reasons, but it is one of the reasons. Why? Because your drug is specifically binding to the desirable receptor, not binding to any receptor. Okay, so if your drug specifically bind to the desirable receptor, then it will produce the more efficacy and less non-specific effects. In other situation, if your drug binds to your desirable receptor but simultaneously binds to the other receptors as well, then what will happen? Your drug will also produce the effects because of the non-specific binding. And that is what we call the adverse effects or side effects. Okay, which is not desirable. Okay, any drug expected to be very specific with least side effect and adverse effect. And that is why the specificity to a particular target of the receptor is really important. And that can be ensured, that can be studied by protein engineering. Then reducing the immunogenicity of the protein. Okay, that is another application of the protein engineering. Then mutation in the drug discovery. So I told you that earlier we had only insulin from the animal origin. Then we switch from animal origin to the recombinant DNA technology origin. But we had only human insulin produced by by what you call by recombinant DNA technology. Okay, and that was the only one form of the insulin. Okay, but later we studied the insulin structure, then established that there are two chains, chain A and chain B, and there are different amino acids and sequence of amino acids, and each amino acid has a specific role. Okay, and when we got to know, then we played with the structure of insulin. And in May 1996, Lispro insulin was what you call developed, where switch B28 and B29 of proline and lysine reduce the association affinity by 300-fold. That resulted in a faster uptake and action as well as shorter half-life. See, why it is required that your insulin should be rapidly acting and should have the shorter half-life. For example, when we take a breakfast, maybe lunch and dinner, so at these three times, your sugar will what we call shoot up. Okay, so but other time your sugar will be maybe in a plateau phase because we are not taking any meal. So in diabetic person, the breakfast, lunch, and dinner sugar or glucose should be covered or reduced rapidly. So when you take just before the breakfast, before that glucose rises, your insulin takes over it and not allowing the peak. Okay, so that is why its rapid action is required. Now, if this is what the importance of faster action. Now, why shorter half life? So you have a space between the breakfast and lunch. Your requirement is that your breakfast sugar should be covered. Now, once it is covered, your insulin should stop its action. Okay? Otherwise, continuous insulin action will decrease the blood glucose very sharp and will lead to the hypoglycemia. Okay? And that is not desirable. So, what is desirable? Immediately after meal, whatever the sugar rises should be taken care, and other time it should be maintained at a maybe baseline. Okay, so that is what the insulin lispro does. So because of the change in B28 and B29 amino acid in a chain B, we could get a faster acting insulin analog, what is called insulin lispro, okay, and it produces the faster action and for shorter time. So this is what the importance of biotechnology in drug discovery or getting the new analog of insulin. Tissue plasminogen activator factor, that is reticulase, a deletion mutant was constructed to reduce the binding of the protein at hepatocytes via EGF domain, that is epidermal growth factor, and that increase the half-life of the reticulase of 13 to 60 minutes and five-fold activity. So this is how the small mutation change, intentional change lead to the increase in half-life and increase in activity. So this is what is possible only because of the biotechnology. Now, post-translational engineering. So post-translational biosynthesis is the covalent attachment of a chemical group and chemical group uh, attachment of a chemical group which is not mandatory glycosylation but attaching the fatty acids or PEG chains alteration of a pre-existing post-translational modification. This post-translational engineering mostly carried out in a CHO, Chinese hamster ovary, and baby hamster kidney cell lines. Then in plant systems such as carrot cells, then Saccharomyces cerevisiae, and what you call various areas, this post-translational engineering is used for a betterment of the process, betterment of the species. Okay, then PEG glycation or gylation, that is alters the physical, chemical, and biological profile of the protein. Okay, so this engineering, post-translational engineering, is another approach to change the physiochemical properties of the proteins. Now, these are the few more applications in of biotechnology in producing the natural products. So natural products obviously can be antibiotics, can be drugs from the microorganisms. So use of mostly actinomycetes as a strain for natural product synthesis. So, for example, use of E. coli for production of 6-MSA or erythromycin and for isoprenoid synthesis. And these are the various antibiotics, okay, or drugs produced in different microorganisms. This is another the application of the biotechnology. Now, biomarkers in drug discovery. So biomarkers, yes, what is present in the body, that is biological, and marker means to recognize, to stage, to diagnose. That is what the marker. Okay, so biomarkers can be used in diagnosis of the disease condition. Biomarkers can be used whether your treatment is effective or not. Biomarkers can be used at what stage the disease is. Okay, so let me give the example of HbA1c, glycated hemoglobin. Okay, so glycated hemoglobin is used to diagnose for diagnosis of the diabetes. The person is a type 1 diabetic or type 2 diabetic, or the healthy person. Okay, then once you confirm that person is diabetic, the doctor will start the treatment. Now, whether the treatment is effective or not, and if it is effective, how much it is effective. You can again measure the HbA1c. If it is less than 7%, that means your treatment is exactly as per the requirement. Even after the treatment, the glycated hemoglobin is more than 7%. That means the treatment has to be changed, or treatment dose has to be changed. Okay. Now, if person's HbA1c remains higher even after the treatment, that means the person is resistant to the oral hypoglycemics, or the oral hypoglycemics are not working optimally. So this is how the one biomarker can be used in diagnosis, can be used to decide the treatment, can be used to check the efficacy of the treatment, and can be used the staging of the disease. Okay. So, for example, rheumatoid arthritis factor, again for diagnosis of rheumatoid arthritis, and there are various cancer markers that can be used for diagnosis, for staging. Then which type of the cancer it is, whether the triple negative breast cancer, or whether there is a role of the BRCA in this, or in case of non-small cell lung cancer, whether the oral chemotherapy, that is erlotinib, gefitinib, this drug EGFR activators, whether work will not, that can be tested by measuring this EGFR mutation. So EGFR is again the biomarker. Okay. So biomarkers are a critical component of the modern drug discovery project. They help you to know the severity and progressive pathway of the disease. They also help you to bridge between the animal models and patients. Okay, and it helps to assess the impact of the development candidate on the intended target. Change in biomarker in response to the therapeutic target can be assessed by measuring the biomarkers. For example, for diagnosis of the myocardial infarction or the heart attack, as per the WHO definition, person should have a typical chest pain. There should be elevation of the blood markers, that may be creatine kinase myosin band, that is CKMB, or troponins. And then third, that is there should be a ECG change also. So if all three changes are there, then you can call that person is having the myocardial infarction. So in that, the CKMB or troponin is the biomarker to identify the myocardial infarction. Okay, when the biomarker indicates complete target inhibition, but the disease still does not respond, it is likely that target is inappropriate for the disease. Understood? So this is what is the importance of biomarkers in disease identification and studying the effectiveness of the treatment in that disease. Now, these are the various drugs discovered using the biotechnology, like insulin. You have Humalog, No Rapid, Gen Insulin R, and these are all for treatment of diabetes. Then factor 8, which is from a Bayer company, that is for males suffering from the hemophilia A. Then factor 9, again from the same company for hemophilia B. Then you have the human growth hormone for growth hormone deficiency. Then erythropoietin, again for like the brand name available in the market, that is Eprex and Epogen, which is used in treatment of anemia. Then a few more important, that is tissue plasminogen activator. The brand name is Activage, which is for dissolving the blood clots. So in this case, we have the three terminologies, that is anticoagulant, antiplatelet, and thrombolytic. So anticoagulant means uh, to decrease the viscosity of the blood or to inhibit the coagulation, you use the anticoagulant like heparin. Heparin again is a biological product, and the synthetic molecule, that is warfarin, coumarin-based drugs. Okay, so these are anticoagulants. Now antiplatelets like clopidogrel, aspirin, these are the antiplatelet drugs. Means they inhibit the activation of the platelets and inhibit the aggregation and adhesion of the platelets, that is antiplatelets. Then you have tissue plasminogen activator, which falls under thrombolytic category of the drugs. Means thrombus is already formed. To lyse that, to break down that, you use the thrombolytic drugs. Now, what is relevance? Anticoagulants can be anticoagulants and antiplatelets can be given prophylactically before actually person gets any myocardial heart attack. Okay? But thrombolytics can be only given after getting the heart attack or getting the evidence that person is having the active thrombus. Okay? So one is after the situation, and the others can be given prophylactically before the actual event happens. Okay, that is what is the difference. Okay, then another application of a biotechnology in actually treating or customizing the therapy, that is precision medicine. Now, what is that? It's basically use of information and data from the patient's genotype and phenotype based on the level of gene expression and or clinical information. Okay, to stratify disease, to select medication, to provide therapy, and to initiate the preventive measures that particularly suited to that patient at the time of administration. Okay, what I mean, to stratify the disease means you can measure the GAD antibodies titer, and measurement of this GAD antibody titer can help you to differentiate whether person is having the type 1 diabetes or type 2. Why type 1 diabetes is an autoimmune disorder where antibody titer of a GAD will increase. In type 2, you will not see the presence of GAD antibodies. Okay? Now, once you confirm that there is a presence of the GAD antibodies and you identify it as a type 1 diabetes, not the GAD and not only GAD antibodies are used, but there are other parameters also to find out whether type 1 or type 2. But yes, GAD antibody is a major parameter to differentiate the type 1 and type 2. So once you differentiate that it is a type 1, then we can choose the insulin as a medicine. Okay? And then you can initiate. So if any person is having the what you call expression of particular genes, presence of the GAD antibodies, then you can take the immunosuppression therapy and prevent, not prevent, delay the onset of type 1 diabetes. So this is how the precision medicine or personalized medicine can help the what you call patient. Now, this precision medicine is basically combination of medicine, genomics, and informatics, okay, to personalize the healthcare or the treatment. Now, this is just a cartoon to explain that earlier, doctor used to see the patient, ask some questions to record the patient history, and based on that, diagnosis used to made, and then treatment used to decided. But now, it's not only sign and symptoms, but the genetic information is also recorded to decide what is the problem, what is the best suitable treatment for that patient, and how it will be effective or specific to that patient. Okay? Now, this genomics data can be helpful to the normal person, to the sick person, and even in a sick person for better disease management. How? In a healthy person, you can check the early markers of the disease and get to know that how that particular person will have the risk in the future for that particular disease. Okay? So you might have heard about the Angelina Jolie, that is Hollywood actress, who massacred her both breasts. Why? Because her genetic testing and the diagnosis told or indicated that she have a high risk of having the breast cancer because of the hereditary or family history. Okay? And that's why she took a in advance preventive measure. Also, in a what we call healthy person, you can test the predisposition markers and find out that at what percent that person will have the risk for particular disease. Okay? Then common and complex diseases can be studied in advance. Okay? This is the application of precision medicine in a healthy person. In sick person, diagnostic markers can be measured and can help the classification of the disease. Okay? And once we rightly diagnose, rightly classify, see, there are three types of breast cancers. One of them is a triple negative, which is the most severe form of the cancer, breast cancer. So once we know, based on the precision medicine biomarker studies, okay, that the person is having the triple negative breast cancer, you will be starting the specific approach of the treatment. Okay? And most likely because of the specific approach, the person's survival will be longer, success rate of the therapeutic outcome will be better. Then, in case of the disease management, as I told you about the HbA1c example, you can test the efficacy of your treatment. You can test the toxicity, okay, and then patient stratification of the what you call for clinical trials. Okay, what I mean by patient stratification in the clinical trials. So you know about this monoclonal antibody drugs. Okay, uh, then there are some synthetic drugs also. Uh, these drugs are effective in only certain type of patients. Okay, and it is because in that patient that gene is mutated, and that is why this drug is effective. Okay, now when sponsor can decide that the sponsor is recruiting only patients who are having the mutation of that gene. Okay, and when we study the drug in such selective patients in a clinical trial, what is the outcome? That your drug under investigation will be effective or not effective. And if it is effective, that means in such patients, it will be even effective and will remain effective even if the drug enters into the clinical stage or the in the market. So by precision medicine, you are excluding the patients who are resistant to that drug. Okay, you are only taking the patients who are sensitive to that drug or responsive to that drug. And that is why you can increase the success rate of the clinical trials. Otherwise, if you take the responsive as well as the resistant patients in the your clinical trial population, then efficacy of that drug under investigation may be different, and that may lead to a different decision by the regulatory agency. So this is how the stratification can be done in even clinical development of the drug. So to summarize my talk, biotechnology plays a very crucial role in drug discovery from the beginning to the end. Okay, even in a discovery stage, even in a what you call development stage. It has a significant application in drug targeting and target validation. In high throughput screening, biotechnology provides really sensitive and flexible method of detection of what you call effect of the drug. And as I said, it is useful from the early stage to the late stage. That means biotechnology covers the major areas of world pharmaceutical companies and major portion of the drug discovery and development. And biotech drug covers a large sector of pharmaceutical market. In fact, you might be aware that in FDA there are two what you call sections, CDER and CBER. Okay, Center for Drug Evaluation and Research and Center for Biologicals Evaluation and Research, CBER. So biological drugs are separately regulated by FDA. That itself is an indication that biotech-based drug or biological drugs have a major going to have a major role in future. In fact, currently they are already showing the importance and their impact. So with this, once again, I'm thankful for patientful hearing and listening to my talk. And once again, thank you the organizers for inviting me and providing me opportunity. So I'll be most happy to answer if anyone have any question.
Thank you, sir. Thank you so much for your stimulating speech. It feels great to have you amidst us. Now, as told, the session is open for discussions and questions. If any of you have any queries, you can unmute yourself or post your questions in the chat box.
Dr. Nandave,
Yes.
this is Satyabhama. Yeah, this is the elaborate presentation which you have given. So it's an enlightenment session, and all the students very interested. But unfortunately, they're not asking any questions. So to start with, I will ask. A, I need a clarification as we are in the need of the hour for vaccination against this COVID. So, what is your perception about the vaccination? It is should everybody should take the vaccination, or we can just contract the COVID and we will gain immunity, that is herd immunity. So, what is your perception about this?
See, if we totally rely on herd immunity, then we would have not having any patient or not that much number of patients in India in the last year. Okay, so that indicates that herd immunity have a role, but not to that extent that it can keep you free from the any COVID infection. Okay, so it's better you should have a
Okay.
vaccination. In fact, whoever is eligible for vaccination, please take it because if all the health workers are on priority vaccinated, and imagine if it is not effective or not safe, then your front warriors will be adversely affected. And you will not warriors to protect you in the COVID vaccine, COVID infection. So if they took the initiative and got vaccinated, that itself shows that the scientific community or the healthcare community believed in the science, and that's why they have the confidence in the vaccine, their efficacy and safety, and that is why they got all the vaccinated. So we go by fact, we go by the evidence, and we should get vaccinated as early as possible, as per our religion.
Yeah, okay, sir. Thank you so much for the clarification. And also, you have given a insight about the biotechnologies aspects. I mean, you have interrelated the pharmacology and biotechnology. So that will be of help to our students. So now I will hand over the session to Dr. Rashmi. So she will just deliver a thank you note to you. So she is one of our colleagues. She is a faculty of the Department of Biotechnology. And if anybody has any questions or queries, the session is open. You can just interact with Sir. Okay, Srijita Nath has posted a question. Sir, what are your views on scope of biotechnology in drug development, especially after the global pandemic caused by COVID-19? Will occurrences like these become more frequent in the near future? So that is her question.
Yeah, Srijita, you asked very relevant question, and as I told you, if you follow the financial budget of India
நீங்க
now,
தேங்க்யூ நோட் சொல்லிருங்க.
if you follow the Indian financial budget, then the allocation of the funds for startup, for healthcare, for pharma industry, and is increased by multiple fold all of a sudden. Okay, that means that in fact you might have heard through your WhatsApp university also that not any temple, not any what you call NGOs or what you call these religious places are helping the COVID patients. Who are helping? The medical science, paramedical science, pharma company, biotechnology, and that itself proves that biotechnology and health sciences will have already having the bright future, but the importance will increase more in the future. And so to support that statement, you already know the this year's financial budget, and so future is really bright. But it totally depends upon how much efforts you put in. And let me share my own experience. When I passed the 10 plus 2, had the option of becoming the doctor, like Ayurveda, dentist, as well as the chemical engineer, and then pharmacist also. But then uh, I had no background of medical sciences like Ayurveda. So I chose pharma sciences, pharmaceutical or pharmacy, only because whether it is a poor person or the rich person, no one can skip the medicine. No one can. And as we are increasing by number in terms of the population and the disease and the longevity, age is increasing, the age-related diseases will also increase in the future. And the importance of drug will never go down. And when there is a importance of high importance of drug therapy or the pharmacotherapy in the treatment of the disease, then there will be always continuous drug discovery and development. And there is the role of biotechnology, which I already highlighted. So you have a really bright future, but only the caution, the important statement that how much you are valuable to the pharma company or the biotechnology company. Because when we hire even a maid, we expect that the person should clean your utensils, mop your floor so perfect. Now imagine when we get the five-figure or six-figure salary, the pharma company will expect that the candidate should be theoretically sound, should be skillful, should be having the multiple, multi-talented or multi-skilled person. They should have the best communication skill, okay, interpersonal relationship, emotional quotient. So these are the all the expectations. So you as a student should modulate in a such manner that you fulfill the expectation of the recruiter or the employer, and that is why what you call importance.
Thank you, sir. There's another question in the comment box. I'll read it out for you. Shalini has asked if you could comment on the role of adjuvants in COVID vaccine development, sir.
Uh, you know, you have adjuvants in general. Adjuvants have already a role in a vaccine. Why? Because they give the stability. They help you to improve the pharmacokinetic properties. Now, if you see adjuvants increase the what you call efficacy duration. Now, if you see adjuvants are basically added to formulate the vaccine formulation uh, for uh, for improving the pharmacokinetic and pharmacodynamic properties. So that is the precise role of adjuvants in the on I mean in every vaccine, but in particularly yes, COVID vaccine also. Yes, I can see the another question by Darshini, where she asked that the science documentary where I saw a group of people in the US who are even against the vaccines essential for born infants. With the COVID vaccine being developed, what are your views that can help those people understand the importance of vaccine? So, if we talk about the infants, uh, uh, not only in US but even in India, regulatory agencies what you call added the caution note, okay, that it should be used in an adult population, okay, because there is a logic. Why? Because in infants the immunity is not that sound. Even the organ system like lungs, liver, kidney, they are not fully developed, okay. So handling the vaccine may be difficult in infants, and in such what you call population, vaccine may not be what you call safer option. Okay, because this vaccine approval is given in a faster and abbreviated form. मतलब इसका मतलब ये है कि with the limited number of patients, means the vaccine was approved even it is studied for a shorter time in a limited population. Okay, but it is an emergency situation. Okay, and that's why they given the abbreviated or expedite approval. Okay, and they have studied only in adult COVID infected patients. They have not studied into the children or infants. Okay, so if you are not aware, if you are not established the efficacy and safety in infants, taking chance directly into the infants is not an advisable or wise option. Okay, and that is why in not only US but throughout the world, vaccines are not advisable or recommended in infants. It's only because we have not studied in such population. That's the only reason.
Sir, I have a question. So, if it is advisable for the patients who have anaphylactic shock or those who have what to say, having allergic reaction to many drugs, so is it advisable for them to take this vaccine?
See, even the person is having the allergy or showed the anaphylactic reaction, see, the person cannot be allergic to every protein or every drug. Okay, so for example, penicillin is a drug, but everyone is not anaphylactic, showing the anaphylactic reaction. Okay, and that is why hypersensitivity reaction is done. Okay, but in case of COVID vaccine, if it is a proven history, then obviously it will be given with caution. Okay. See, anaphylactic reaction can be managed by epinephrine, by antihistaminics, and steroid drugs. Okay. But if it is proven that so and so person showed the anaphylactic reaction in the past to a particular drug, then yes, it will be decision.
Okay, sir. I need to confirm one message, sir. Actually, I got it in WhatsApp that a person who had anaphylactic shock after taking the person had taken the vaccine shot and she had got myalgia, and she's also a doctor. She has finished MBBS and she's an MS student, and she has got anaphylactic shock after taking diclofenac, and she died within 2 days.
Oh,
So,
yes.
Because I need to confirm this, because as you said, I also had anaphylactic shock before to other medicines, and my doctor said you can just, if you have any anaphylactic shock, we will be treating for that, not for the COVID vaccine. So I just want to confirm whether what is the status of this message, the truth behind this message.
See, I said basically WhatsApp University.
Yeah.
Why? Because everything is not scientifically proven, or not all the messages are composed by or sent by scientific people.
Yes,
Okay?
I didn't want to spread what to say.
So it's absolutely rubbish.
Yes, sir.
See, diclofenac or NSAIDs are proven to cause hypersensitivity reaction. Okay, so it's not because of the COVID vaccine. It may be diclofenac itself. Okay, so there is no what you call question of the anaphylactic reaction to COVID vaccine.
Yes, sir.
The other drug, that is what you said in the WhatsApp, that is diclofenac. Okay.
Yes, I just want to tell to everyone that not the COVID vaccine is causing the anaphylactic shock.
I do not believe in such rumors, and see, there is a lot of commercial competition involved. Like there are so many vaccine candidates from the AstraZeneca, Pfizer, and other companies, and there are Indian companies also. Okay, so imagine we are vaccinating our people and we have capacity to export it also to the other countries. Okay, and if we capture the market of other countries, then it may be adversely affecting the Pfizer or maybe AstraZeneca or other companies. So there is a possibility that is a money game. Okay, that you, rather than you prove your vaccine candidate is more effective, your other strategy can be that you spread the rumors about your competitor and create the bad name. That may be the possibility. Okay, so it's all money game.
Thank you, sir.
Okay.
Thank you, sir. Is there any more question left?
Thank you, sir, for your wonderful presentation.
Mhm.
I just have one question. So you just told about precision or personalized medicine.
Yes.
So even now it is at the juvenile stage, even though we have been talking about this for a past decade, so it is still in the juvenile stage. So what is your opinion on this? So why is it, why are people not aware of this particular thing?
Uh, See, it's not about like. In fact, personalized medicine is already. Let me give very known example to me that one of the relative had the non-small cell lung cancer, which is the type of lung cancer more common in females who are non-smokers. Non-small cell lung cancer is common in females who are non-smokers. Okay, so in such a type of lung cancer, I'm talking about like maybe 10 years before. 10 years before. You are talking in 2021 that why people are not aware. I'm talking the 10 years before the situation. So that person started with the parenteral chemotherapy with the paclitaxel and other drugs. Okay. But then after 7 chemotherapy cycles, she started showing very adverse reactions to the parenteral chemotherapy. Now, cancer specialists decided to shift her from the parenteral chemotherapy to the oral chemotherapy. Now, in such a type of cancer patient, oral chemotherapy option is EGFR activators. Example, JP-TNIB, erlotinib. There are so many TNIB drugs. So JP-TNIB is the number one drug in that, then erlotinib. So to shift from the parenteral chemotherapy to the oral chemotherapy, doctor has to ensure that there is a EGFR mutation in such cancer patient. So the patient sent the sample to the diagnostic lab and then study that there is a mutation. Okay, and when there is a mutation, then doctor shifted her from the parenteral to the oral chemotherapy, that is JP-TNIB. That indicates that doctors were able to only shift her from the parenteral to the oral chemotherapy because that patient had the EGFR mutation. Okay, so this is 10-year back example, and which is very known to me. Okay, so that means this personalized medicine or precision medicine is already existing and is used or practiced. Another example, the first line chemotherapy, that is, first line anti-tuberculosis therapy includes the rifampicin. Okay, so in rifampicin, you have two types of Indian tuberculosis patients: fast acetylators and slow acetylators. इसका मतलब ये है that rifampicin can be metabolized faster in some patients and can be metabolized slowly in some patients. Okay, so why it is fast and slow acetylators? Because acetylase is the enzyme which metabolizes this rifampicin into inactive metabolites. Okay, so if it is faster metabolized, then rifampicin will be rapidly cleared from the body and will have the less anti-tuberculosis effect. If it's a slow acetylator, then the drug will be slowly metabolized, will have the more effect. So when you stratify who is the fast accelerator and who is the fast and slow accelerator, depending upon you can give the dose of rifampicin. The person who is a fast accelerator will give the higher dose. The person who is a slow accelerator will give the small dose. Understood? So these are the already known. So this is what this is already practiced in a clinical practice. It's not a
Thank
use.
you. Yeah. Thank you, sir.
Mhm.
So there is one more question by Vaishnavi.
Okay.
Yeah. So, what is your view about booster vaccines? Many people avoid them, which makes their vaccination chart incomplete. Is that important to have boosters? So, this is a similar kind of question, I suppose,
Yeah.
which was asked.
This
Yeah.
itself indicates that to get complete protection and to get the complete antibody titer, you need a booster dose. Okay, and yes, avoiding them is not the option. Taking them is the right approach. So yes, booster have a major role to keep the efficacy, optimum efficacy of that particular vaccine. So everyone should take the booster dose if it is recommended. So in COVID also, that if the person take the first dose on the 10th March, then after 28 days, the person should take the second dose. The plasma therapy is not a vertical specific approach. Is a just a non-specific way to manage. If nothing was there, then we tried with the azithromycin, we tried with the steroids, we tried with the analgesic antipyretics, we started with the plasma therapy. But now we know that particular antiviral drugs are required and a particular vaccine is required.
Thank you, sir. Now, this is for the attention of all the participants in the meet. The link for the feedback form has been posted in the chat box and it's also been being circulated in the WhatsApp group. Please do fill it up. And now over to Rashmi, ma'am.
Thank you, sir, for your wonderful talk. So it was really useful, and hope all the students would have got benefited by your lots of information and lots of inputs from your side. Thank you once again, sir, for sharing your valuable time with us.
Yeah, it's my pleasure. And if anyone need any help, I have already given my email ID. You can write to me at mukeshnandu@gmail.com.
Yeah, thank you once again, sir, for your valid inputs and promptly accepting our invitation. Thank you so much.
Yeah. So I have another meeting. So can
Okay.
you know?
Yeah, we can wind up, sir. Yeah,
Okay.
thank you.
Thank you so much, and thank you to principal and all the organizing committee members. Thank you very much. Thank you,
Okay.
Dr. Rama.
Thank you, sir.
Now it's time for a short break. Please be back after 10 minutes. Thank you.
The session is going to resume by 1:30 p.m. after lunch. So I hope to see you all by that time. Thank you.
ஹலோ மைக் டெஸ்டிங் கேக்குதா? மனுஷர் அன்பே மனுஷர் ஓகேவா சார்?
Dear participants, good afternoon to everybody. And whatever you are speaking, everything is recorded. And we are recording this conference from the morning. So I request you to maintain silence and put your mic in mute and also switch off your videos. Just listen to what the speaker is telling. I request your cooperation for the smooth conduct of this conference. So we have attained the maximum limit in this Gmeet. So you can tell your friends to join in the YouTube link. So the YouTube link is given in the chat box. So you can just send that to your friends and they can join watching the YouTube.
Good afternoon, ma'am. நான் மியூட் பண்ணிருக்கீங்க மேம். மியூட்ல இருந்து வெளியிடுங்க மேம். மணியர்
மணி சார் வாங்க சார்.
சார் சீக்கிரம் வாங்க. மணி சார் கூகுள் மீட் ஒர்க் ஆகல சார், சீக்கிரம் வாங்க. மணி சார் Hi, good afternoon, everyone.
அப்படியே மனப்படம் பண்ண
Jenny. யூடியூப்ல இல்ல, இதுல பேசுறேன். யாரு?
இப்ப நீ காட்டுதா? அந்த ஹியூமன் அந்த கடைசி ஒரு மனுஷன் டேனியல் கேம் விளையாடாத.
Good afternoon, madam.
Good afternoon, sir.
Good afternoon. Okay.
Sir, just a moment, we will just start.
Okay.
Hello, everybody. It's good to have you back. Now, let's get into the next segment.
Now I call upon Ms. Satyabama, Assistant Professor, Department of Biotechnology, to introduce the second speaker of today's session. And please do the honors.
Good afternoon, all. I welcome Mr. Ganesh, who is the founder and chairman of the Pondicherry Institute of Agricultural Sciences, Pondicherry, for the second session of this national conference. And to give a brief introduction about Mr. Ganesh, so he has given his resume like the way of development of a nation passes through villages. So it's quoted by Mahatma Gandhi ji. So he has said like the quotation strikes me, and after visualizing the actual picture of rural India, he has decided to devote the professional career mostly for the development for the rural society and the rural people. So as a first step, he went to Mozambique, that is South Africa, during the year 2002 for introducing black gram, which is the pulse crop cultivated in that country. So he has given the technology to the country and helped thousands of farmers. So returned back to India and attended the agri clinic and agri business training by the Ministry of Agriculture, NABARD, and managed trusts. So during the year 2002, so he was in the Green Tech Horti consultation. So it was the first ever in Pondicherry by agri graduate, and he was also involved in consulting many agro industries and agro farmers. So through this Green Tech Horti consultancy firm, he has promoted many commercial agricultures like polyhouse consultancy, organic farming, orchard, vermicompost production, etc. So in 2003, he started the SEED, that is Center for Environmental and Agricultural Development, that is an NGO at Pondicherry. For helping the farming communities and to empower women. So through SEED, he has executed many projects in sustainable agriculture, soil water conservation, livelihood promotion programs through NABARD, DSTE, Ministry of Agriculture, and the Department of Tourism. And for his honor, he has started the Pondicherry Institute of Agriculture Sciences in collaboration with Annamalai University during the year 2017. So as you all know, it's offering diploma programs in agriculture and horticulture, and also the MBA program in agri and food business management. So he is the recipient of many awards. So to name a few, the first award is the Innovative Entrepreneur Award from Indian Council of Agricultural Research, New Delhi. Next is the Social Entrepreneur Award from World CSR Forum, Mumbai. And third is the Kamal Patra Award from JCI India, and Best Farmer Award from the Right Channel, and Best Agri Entrepreneur Award from All India Agricultural Students Association, New Delhi. And apart from this, helping the research. So for his credit, he has 11 popular articles. He has 5 research articles and books and manuals and also abstracts. So he has a total of 30 publications, and he is also a media person. You can often see him in the Makal TV, Kalinyar, Tandi TV, etc. So this is a brief introduction about Mr. Ganesh, and I hand over the session to Mr. Ganesh. We welcome you, sir.
Thank you, madam, for your introduction. First of all, good afternoon, everyone. Pondicherry Institute of Agricultural Science, and full thanks to PSG College of Arts and Science for give an opportunity to share our views about the impact of COVID-19 on agriculture. And this seminar, we are going to present about the impact of COVID to agriculture. So first of all, our regards to Department of Biotechnology from PhD College of Arts and Science for organizing this 2 days national seminar on recent trends in applied aspects of biological sciences. And in this seminar, we are going to do a presentation about the impact of COVID-19 on agriculture and also an urge to novel development from our side. Before, let's go to the presentation first. Then we have a short discussion at the end of the program. Thank you very much for a nice introduction about the PIAS. It was started in the year 2017 in support of Center for Environment and Development. Since 2003, we are working for the farmers and women empowerment in coastal region of Pondicherry and Tamil Nadu. In 2017, we have started this institute called the Pondicherry Institute of Science. We are offering diploma courses and certificate courses with the help of Annamalai University. We have conducted 3 international conferences in the last 3 years with the help of other institutions. So with this background, we are going to present about this similar topic of impact of COVID-19. This time last year, concept of such as lockdowns, mask mandates, and social distancing were unknown to most of us. Today, they are part of our everyday language as the COVID-19 pandemic continues to impact all aspects of our lives. Over the past 12 months, the pandemic has harmed the poor and the vulnerable the most, and it is threatening to push millions more into poverty. This year, after decades of steady progress in reducing the number of people living on lower income, COVID-19 will usher in the first reversal in the fight against extreme poverty in a generation. As I told, the COVID-19 induced lockdown in India was huge economic shock. It started across the country on 24th March 2020 and is still ongoing with the restriction in one form or other. It stalled the economy with a complete closure imposed on the enterprises across all sectors, including agriculture. Even though agriculture activities were exempted in the initial phases of the lockdown, the agriculture value chain also faced large-scale disruption. This had a serious detrimental effect on the rural economy. The coronavirus pandemic has also triggered a massive reverse migration from the urban to rural areas in large part of the country. The times of the crisis is a greater teacher as long with the inherent challenges, it can throw open many new opportunities. In the present coronavirus pandemic also, the immediate challenge was restoration of the supply chains of the essential commodities as well as reducing the plight of the distressed migrant worker. The government, though its various interventions, specifically through the Prime Minister Garib Kalyan Yojana and the Mahatma Gandhi Rural Employment Generation Program, has provided timely relief to migrants in these difficult times. While most of the challenges presented by the pandemic have been efficiently handled, it is also important to make use of the opportunities that crisis provides. The importance of agriculture, actually, India is home to about 120 million smallholding families who contribute over 40% of the country's grain production and over half of its fruits, vegetables, oilseeds, and other crops. Much of the global share of the food staples such as rice and wheat come from India only. And almost half of the population in India depends on agriculture for their livelihood. All this every year, the Indian farmers face risks such as low rainfall, drought, price volatility, and rising debts. Including these invisible disasters, there are some other risks, but risks from the COVID-19 pandemic are putting new challenges in front of the sector that is already under the threat. In seeing this presentation, actually, this slide shows that we are the largest producer of the milk, pulses, spices, and tea and jute. We have arable land, second largest arable land in the world, almost covering 7,500 kilometers in the coastal line. Whereas the second largest producer of fish, cotton, fruits, and vegetable, poultry. 58% of the livelihood depends on the agriculture. And 44% of the country's workforce employed in agriculture. This is a fact about the agriculture scenario. Although India is one of the largest producer of some of the agriculture and horticulture products, yet the national yield of major crops, except groundnut, is less than the global average yield production. Further, the national yield of such crops is far less than the highest yield achieved in other parts of the world. In addition, the COVID-19 pandemic has adversely impacted the global agriculture sector, particularly Indian agriculture sector. The major impact of this agriculture is first on production. Impact on production is at the all India level, agriculture production in almost 47% of the districts are adversely affected by the impact of the COVID-19. Magnitude-wise, agriculture production is minus 2.7 and not been adversely affected significantly, mainly due to the fact that harvesting of the rabi crops like wheat was almost complete by the end of the April 2020. That is the harvest season for the rabi season. However, the production in allied sector had declined significantly, especially in the poultry sector, minus 19.5%, followed by fishery sector, minus 13.6%, and sheep, goat, piggery sector, minus 8.5%. Primarily due to the drastic decline in the demand for this product, possibly due to the widespread fear circulating in the wake of the COVID-19. Because of the lockdown and the people are not going outside, there is no purchasing power in that time. Also, production in dairy minus 6.6% and horticulture minus 5.7%, going to the reduced demand for these products and the disruption in their supply chain. This is a major impact on agriculture, especially in production side. Whereas impact on farm gate prices have not declined significantly in the crop sector, minus 2.2 only. However, the prices in allied sectors have declined in the range of 2% to 18%. This decline was highest in the poultry sector, almost minus 17.8%, followed by horticulture, minus 1.6%. Mainly due to the supply disruption caused by the restriction on movement of vehicles, is a major reason for the shortfall of prices in the agriculture sector. On the whole, 54% of the sample districts have witnessed adverse impact on the farm gate prices of agriculture produce due to the COVID. The next impact is on availability of agriculture imports. The major agricultural inputs like seeds, fertilizer, pesticide, fodder also have heavily impacted due to the COVID-19, due to the restriction imposed on the movement of men and material during that time, and also closure of the shops, availability of agricultural inputs, these seeds, almost minus 9.2%, fertilizer minus 1.2%, pesticide minus 9.8%, and fodder minus 10.8%. Decline in the range of 9 to 11%. At all India level, 58% of the sample districts are adversely affected in terms of the availability of inputs due to this COVID-19. Another major impact in agriculture is impact on prices of agriculture produce. Price also sharply decreased due to the disruption of supply chain owing to the restriction on movement of vehicles and the closure of shops and markets. Prices of agriculture pesticide inputs like seeds 8.8%, fertilizer 10%, pesticide 8%, drastically reduced. At all India level, 54% of the sample districts are witnessed an increase in prices of the agricultural inputs, possibly due to the non-availability. The major reason is non-availability to get the higher price. Impact on agriculture marketing. Even though local procurement centers were opened by various state government under their jurisdiction, yet restriction on movement of vehicle had adversely impacted about 75% of the sample districts in smooth operation of the agriculture marketing through this. The impact on operation of the rural HATs was most severe, with 87% of the sample districts being adversely affected. This was mainly due to the complete ban on opening of rural HATs by the local authorities in majority of the districts in the country. So other impact on agriculture is impact on banking services. It is indirectly related, directly related to the farming sector. As far as the banking services are concerned, access to credit through term lending and KCC, Kisan Credit Card, was adversely impacted in about 89% and 59% in the most of the districts in India. As regards to the recovery, 94% of the sample districts were reported to have been adversely affected by the pandemic and the consequent lockdown. However, a positive feature that emerged was that almost 63% of the sample district reported an increase in the digital transaction by their customers during the lockdown period. Other major breakthrough in new technologies, most of the people are used to have their digital transaction due to the COVID-19. The impact on MSME sector is also related to most of the farming related to micro, small, medium enterprises. This also affected because of the COVID. MSME sector was the worst hit sector by the COVID-19 pandemic in terms of impact on price level of raw materials, employment, production level, consumer demand, and disruption in the supply chain. A decline in production level and employment was reported in 97% and in 96% for the sample districts respectively. Similarly, adverse impact was reported on consumer demand and the cash flow of MSME sector, thereby increasing the heart of people at the large level. All this agriculture sector was affected by the COVID-19. Government have taken many steps for the mitigating the ill effect of this COVID-19. The response of the Indian government in agriculture has involved 3 policy measures. One is that it has announced that most agriculture activities will be in essential list. It has exempted farm workers in the field, farming operation by farmers, agencies engaged in the procurement of agriculture products, including MSPs, mandis notified by the state governments, inter-intrastate movement of the harvesting and sowing related machines and manufacturing, packaging unit of fertilizer, pesticide, and seeds, among the others. It's a one steps initiative taken by the government of India in that time. Second is that it has announced that the first installment of Prime Minister Kisan payment to the farmers, that is rupees 2,000, will be paid upfront to the farmers, all the farmers. It has also announced that the wage under Mahatma Gandhi National Rural Employment Generation Program will be raised from the rupees 180 to 202 per day. It also significantly helped the most of the vulnerable community in that period. Here, initiative taken by the government of India is the Reserve Bank of India has announced a moratorium on agriculture term loan, including a crop loans for a period of 3 months and extended later. Okay, let's see the way forward for after the significant impact of the COVID-19 on agriculture. COVID-19 has exposed the vulnerability of India's agri-food systems and accentuated the need for agriculture market reforms and a digital solution to connect farmers to markets, to create safety nets and ensure reasonable working conditions, and to decentralize agri-food system to make them more resilient. For example, this agricultural food chain is connected to depends on agriculture inputs like seeds, fertilizer, crop protection activities like pesticides, farm equipments like machineries like tractors, implements, irrigations like micro irrigations, drip sprinkler, insurance also, agriculture insurance. These are the critical inputs for the agricultural development. In the value chain, these inputs are necessary to get farming, like food grain production and horticulture development, commercial crops through polyos technologies, and oilseed production, especially groundnut and other major crops like gingerly, sunflower, and in addition that allied sectors of agriculture like livestock and animal husbandry and poultry also involved in the farming activities, essential, and also fisheries also another important sector in the farming sector, where is in the part of the value chain. After the COVID, most of affected value chain and in the system is warehousing and logistics systems affected abruptly during that period, whereas curfew and stoppage of this transportation leads to the most of the agricultural produce are left in the farmer's field itself, result in the huge loss to the farmers and also to the economy. So there is a need for a processing of these all these product is very essential to create awareness and also to capacity building of the farmers is essential in the process sector. Like we need to develop cold storage and food processing or value addition, and a proper logistic is essential in this food chain system, the value chain system. And also, it is very essential to have market facilities to link all the produce produced by the farmers in the small stores or mega malls like Reliance, mega malls, unorganized sectors, vegetables shops, or whatever Sunday like need essential need importance to create the marketing of all these agriculture products. Okay, what's the future growth in agriculture after this COVID-19 is that now smart agriculture is developing in the sector of agriculture, where subsistence farming or sustainable farming is now converted into smart agriculture system, like also is a precision farming system. Collective farming is another area where farmers are involving after the COVID-19. High value agriculture production products also essential to mitigate the effect of pandemic situation in future. Food processing industry also play a vital role in the future growth of agriculture. In smart agriculture, this slide shows the how the smart agriculture play a major role in the development of farming sector in future after the COVID-19. It's one of the labor-saving technology, and also a large-scale production is possible by this smart farming technology. Now drones also used this type of after the COVID-19, most of the farmers are also using the drones, other latest machineries to minimize the labors. It's a good news is that government of India has now increased in its focus on nutrition. Earlier, they concentrated only on the food security only. Now they're focusing on nutritional security because they are now more concentrating on health point of view in the humanitarian concern. So now nutritional based production is more possible in future, and they're raising the farmers' income rather than the enhancing the farm productivity. Earlier, government of India focused on the farm productivity, that means the yield per area. Now instead they want to raise the income of the farmer. Second is that changing the consumer behavior with the suitable programs, the incentives is already in the agenda of the government of India. For all these to happen, the existing landscape of policy incentives that favor the two big staples of wheat and rice has to change. Recently, the rice and wheat is a major crops by farmers. There is a major water consumption crops, and also more inputs are required to produce yield. Now then we need alternate cropping system, alternate method of technologies also in the pipeline for getting the development of the farming sector. Design agriculture policies post COVID-19 scenario must include these imperatives for a food system transformation in India is another initiative taken by government of India. The action plan. Based on the findings of the survey and the feedbacks received from the stakeholders, different stakeholders, few policy suggestions for mitigating the impact of COVID-19 on rural India are followed. First, due to decline in agriculture and allied sector production, direct income support may be provided to farmers in general and those engaged in poultry and fishery sector in particular. In this connection, enhancing the income support through Prime Minister Kisan could be a good option. This is the one such a plan taken by need to be taken for the development of farming sector. Due to disruption in the marketing of agriculture produce in mandis and rural hats and reduced farm gate prices, the income of farmers declined, leading to poor recovery. Therefore, interest waiver for agri term loan for at least one more year may be thought of. This is another recommendation. Microfinance activities need to be reactivated through injection of liquidity to NBFC, MFI, non-financial, banking financial, microfinance institution sector, so that small business activities on pavement and the roadside shops could be circulated in semi-urban rural areas. This is another important point is banks should be nudged to enhance credit linkage under next dose of the credit to eligible SHGs at lower interest rate and interest subvention and the waiver of the interest for at least 2 quarters for existing loans. Opportunity for mask making, sanitizers, direct delivery of the food grains, vegetables, fruits to be encashed by self-help groups and the FPOs, farmers produce organizations, also essential. Next is the awareness program on COVID-19 by self-help groups, farmers producer organization, and the FCOs in rural areas may be done in massive scale to check the growth of the corona pandemic. Universization of Mahatma Gandhi National Rural Employment Generation Scheme for covering more and more laborers, including those migrant workers who have returned from the bigger cities. It's another area where need how to show important is rural grounds and cold storage infrastructure capacities in rural region need to be augmented. And also model farm equipment, that means fodder bank or with the FPOs, farmer producer organization may be developed with support of the NABARD. I think this year Government of India is planning to have 10,000 FPOs to support the farming sector and also for empowering the farmers. Conclusion, the impact of COVID-19 has drawn numerous comparisons, some to the global financial crisis of 2007-8. This financial crisis happened in the 2007-8. Others to World War II, and most still crisis we know only from history books. While these may seem dramatic, the pandemic has had wide-ranging impact on nearly every aspect of development, like few crises before it. So the full scale of the pandemic will only known years to come as we collect and analyze the data, adapt, evolve our financing to meet countries' needs, and continue our end extreme poverty and promote shared prosperity. In the long term, the government should develop more robust export infrastructure, change policies to allow for bigger focus on processed foods, and reduces the government buffer volumes. And also the micro enterprises also to be developed in the area of agriculture, like decentralization of the micro enterprise also very important, where allied activities like animal husbandry to be promoted, beekeeping is another area government is giving important for sustainable farming production to mitigate the COVID-19. Then promoting organic farming through this various sustainable activities, production of vermicompost and other organic inputs in the farm level. It reduces the cost of production and also the increase the income of the farmer, and also the value of the product, the quality of the product is also increasing. Now, farmers as well as government is focusing on instead of quantity production, they have quality production. Earlier, they have more concentration on the quantity production, more production and the more productivity. Now they need to have a healthy, is the important aspect they are concerning now everyone. So these to be addressed is now need of the hour after this COVID-19. And other major changes after this COVID-19 agriculture sector is most of the farm women and also the youth are interested to take self-employment or agriculture enterprises in the homes itself, in the hometown itself. For example, there's a drastic change in the migration of the IT people from other field also to the farm sector, especially in the mushroom cultivation. Most of the people are entering in the mushroom cultivation as a profitable venture. They can do it in the home itself, the smaller area with the latest technology, they can earn a sizable income for their livelihood. In other areas now is a new technology they are now introducing in the agriculture after the COVID pandemic situation, especially usage of mechanization, especially the drone is one such a new latest device and measures using for the spraying of the crops, because the now labor shortage is the other issues facing besides the COVID-19 pandemic issue. So we as the agriculture sector need more give more emphasis on the production side, sustainable production side. This COVID-19 given a lesson to all the farmers. Also, the government also thought about the revitalizing the and renovating the agriculture sector into the new era, where everyone should get sustainable income with maintaining their health. So, with this, concluding this session about the impact of COVID-19 agriculture, a lot happened. A lot of things to be addressed by the government in future. Recently, we are also again hearing the news that again one more lockdown is coming soon. But in agriculture sector, we need more attention for sustainable growth, because it defeats the entire world. So without food, we cannot again run this society in a proper manner. So government is giving more initiative and also assistance to the farmers for the sustainable production of agriculture. So thank you very much to all the participants who are hearing this seminar. If you have any queries, note, please let me know. We have a small discussion about this topic. Thank you very much.
Thank you, sir. Thank you for volunteering your time to be with us here today. I'm sure that your words are resonating with each and everyone present in today's meet. This is for the participants' information. The link for the feedback form for this session has been posted in the chat box. Kindly do take your time out and fill it out. And now the session is open for further questions and discussions. You people can unmute yourself or post the questions you have on the chat box, please. Thank you.
Okay, as there are no questions from the participant side, I would like to interact on a specific aspects which you have just told. You told, like, now the government of India is contributing towards the nutritional value rather than the food value. So in what terms we can get along with it, or what is the strategy of developing a nutrition based crop, or what is the strategy, sir? Can you just explain that?
Okay. Actually, earlier we are concentrating only 2 crops. Okay. Just a minute. Disruption. Okay. Are you hearing? Hello?
Yes, sir, we can hear you, sir. Yeah, yes, sir, we can hear you.
Earlier, we have considered only on the production of the cereals and other major crops in oil seed. Now the government is giving importance on the health aspect, nutritional based production, especially the now the promoting the vegetables cultivation, especially in the kitchen garden. So in the urban areas through rooftop garden, they are promoting such situation. they are giving best food instead of giving more important to only cereals. Now, pulses also play a major role in the for their health aspect of food production.
Okay, sir. Thank you so much for the clarification. Okay. Now the session is open for discussion. So if you have any questions, you can just post it on the chat box, or you can just unmute and you can ask the question of your choice.
Okay, moving on to the next session, I would like to call upon Ms. Niveta ma'am, Assistant Professor, Department of Biotechnology, to thank the speaker of today.
Good evening to all. Thank you, sir, for your presentation. You had explained clearly about the need of agriculture under this period. Smart knowledge, agriculture technology, and also detailed government policy. These informations are very useful for our delegates, and also it will be very useful. Good evening to one and all present here. Thank you, sir, for your presentation. You had explained clearly about the need of agriculture and its value in their pandemic period. Finally, you had explained about the smart agriculture technology and also detailed government policy. This was very useful for our delegates, and also we will think about the in future. Thank you, sir.
Thank you. Thank you very much.
Moving on, now there will be oral presentation held, and I warmly welcome Raja Muthuramalingam for presenting contents based on intrinsic self-assembled plant virus capsid, protein multifunctionalized as a nanocarriers for cancer cell-specific theranostics.
Before that, Darshini and Pritika, our juries didn't join. So let us wait for them to join. They will be joining in 5 minutes. So once they join, we will start out with the oral presentation.
Sure.
Yeah.
Hi. Hello.
Hello.
Dr. Raja, please wait for a moment. We are waiting for the panel members to join. They'll be judging your presentation. So let's wait for 5 minutes.
Fine.
Yeah. You can start sharing your slide. That's not a problem. We'll be just waiting for the judges to join.
Sure.
Okay, here we have our first judge, Dr. Vasant Dakshinamurthy. So Dr. Vasant, he is working in National College Trichy in the Department of Biotechnology, and he has more than 10 years of research experience in animal model studies, and he is one of the judges for evaluating the presentations. We welcome you, sir, to this national conference.
It's a pleasure, Dr. Satya, to join this program.
Yeah, one more. Thank you, sir. Thank you for promptly accepting our invitation.
It's a pleasure.
So one more judge, she has to join, Dr. Pratima. She'll be joining in 2 minutes. So let's wait for her. Then we will go ahead with the presentation.
Sure, sir. Yeah.
Pratima ma'am, are you here? Pratima ma'am. Pratima ma'am, are you here? Pratima ma'am. Okay, we have our second judge also here. So I welcome Dr. Pratima Sambandam, senior scientist, Department of Biotechnology, and she is working in Sugarcane Breeding Institute. She has more than 10 years of research experience, and now she is holding the position of senior scientist. And her area of interest is genetics and genomics. And we have two eminent judges for evaluating the presentation. Dr. Vasant Dakshinamurthy. He is an expert in animal sciences, and also he is working in animal models. And we have another judge here who is a genomics specialist. So over the session to both the judges. We can go ahead with the first presentation.
Yeah. Is that my PPT is visible now?
Yes, doctor, it's visible.
Yeah, thank you so much. Hi, hello everyone. This is Raja Muthulamalingam doing postdoctoral research at Indian Institute of Technology, Delhi. So I completed my master's in Trichy and I completed my PhD in biotechnology at University of Madras. So here we're going to discuss the story about how pathogenic viruses use materials for various applications. So here especially we are going to see for cancer cell specific diagnostics. So diagnostic means detection and treatment, either killing, we can say it like that. So here, so our researchers started domestication of viruses from last century. So the kind of virus vaccines they created, and even most of them are already vaccinated from our childhood days. So they started using viruses as a vaccination even last centuries ago. And recently they started using as viral vectors with vectors like from the nucleic acid, they used adenoviruses and the retroviruses for HIV therapy. So HIV therapy most, you can find that adenovirus therapy is the most possible therapy for HIV treatment. So the people, researchers are used to viruses, domesticated viruses for various purposes. So in material science researchers review, they started using, they found something very interesting and virus particles. So they started using virus particles as a nanotechnology approaches. So here the human being will do anything and everything for research purposes. Even if you think that dinosaurs is available, they will domesticate everything for a human useful purposes. So like that, they domesticated the viruses for centuries ago and recently. So in material content, so people are started using the viruses as a nanotechnology purposes. So how viruses could be a part in nanotechnology? If you see that most of the viruses will come under the size between 10 to 30 nanometers. That's the nanometers range scale we can say. So viruses as a nanomaterials. We have viruses more than 500 nanometer and 1000 nanometer also, but we cannot say that the virus above 500 nanometer is a nanoparticle. We can say it's a microparticle. So from that comparison, so people have started using viruses as a nanotechnology purposes. So what other advantages is in the material science research thinking. So in material science research point of view, the material should be in same size, in homogeneous size. If it is a heterogeneous size of particles, so the application oriented, it will be get negative impact. So they are found that viruses are homogeneous size. Even if you are taken 1,000 viruses of particular specific viruses, all the particle size will be the same. So it is much suitable for the nanoparticle approaches. What the material scientists thinking. So if you thinking about the virus architecture, there is only major 4 or 5 parts is combined with consist of viruses. If you going with a mammalian structure, virus structure, you will find some glycoprotein. Most of the mammalian viruses will have the glycoprotein. Even if you think about the coronaviruses, it will have some glycoprotein, a kind of spike protein, which is used for the diagnosis and other purposes. So on another part, it will have some capsid protein. So capsid is a main structure of viruses. It is contained capsomeres. So capsomeres is made of n number of monomers of capsid protein. So that capsomeres will have the monomers with the structure, and otherwise the amino acid structures and molecular wise it's more stable and more similarity to other capsid protein. So and another part it will have nucleic acid. So only 4 parts will have virus will have the structures like nucleic acids and proteins and glycoprotein and capsid. So later plant viruses, researchers were found plant viruses is very useful for biotechnology applications and nanotechnology applications. Why? Because when we go into the mammalian viruses, most of them are highly infective human beings. So researchers decided to go with the plant viruses because plant viruses is non-pathogenic to human beings because we everyday consuming raw plant material, agri material. So if you taken the salad, cucumber salad or a tomato salad, if you drink some sugarcane juices, if you drink some musubi juices, if you drink some any kind of raw juices, it must contain any of the viruses. So till there is no report of viruses cause any negative impact of human being. So the researchers decided to use plant pathogens for nanotechnology approaches, and also uh, in plant viruses we can easily isolate, we can easily purify in the large quantity. If you go to the field, if you go to the outside, you will find a lot of infective materials. From there you can isolate the plant virus material. So in the ease of plant virus material, so most of the researchers started doing with plant viruses. And here you can see the one of the picture is a German young woman scientist. She started with the plant virus nanomaterials. His name is Nicole Steinmetz. He's just very 30 years old. So from on the day itself, most of the researchers have started doing. In India, I can proudly say, I was the one started with the plant virus nanomaterial for nanotechnology approaches. So here you can see the virus nanoparticles. So still date we have the researchers are identified more than 1,500 virus particles, and only the plant viruses are used for the nanotechnology approaches. the size and shape we can say icosahedral, almost like spherical size. In another size we can say the rod shape. So these are the virus particles researchers started using. Already there is a lot of product available which is based on the virus nanoparticles. Also, we can say that viral-like nanoparticles, which means viruses only with capsid protein without nucleic acid. They will say the virus-like nanoparticle. If you want to create the virus-like nanoparticles, you have to synthesize. You have to do some recombinant engineering to produce the monomers, which is self-assembled, intrinsically self-assembled to a virus-like nanoparticle. It means it will form like a nanocargo-like structures. I mean, real virus-like structures without nucleic acids. From which with the virus-like nanoparticles, we can use some external molecule or internal molecule to conjugate for the biomedical applications. For example, if you want to conjugate viral nanoparticle, if you want to encapsulate viral like nanoparticle with Go nanoparticle, magnetic nanoparticle, quantum dots, even foreign DNA, it can be encapsulated in the viral nanoparticles that we can go for the further applications. So it is very easy to comparatively very easy to recent technologies. So this is my experiments on VLP. So I started during the middle of my PhD. I started when I read that book of Nicole F. Steinman. So I started working with VLP. Even in the middle of my PhD, I just put my previous work. I started newly with working with the VLP. So I started collecting the samples in the field samples. So in field samples, in symptom based, I collected. The symptom is you can see a lot of symptoms in the field like necrosis. There is a major symptoms for any plant infections and wilting, standing. So based on that, I collected samples and these viruses are transmitted by white light. That picture, electron microscopic picture, in red color. Also, you can see that white fly is a transmission used for transmission of viruses to infected plants to healthy plants. This is our first report from the particular specific viruses. So here I used ultracentrifugation for a purification of virus particles. So in ultracentrifugation is the best technique to purify the virus particles. So we will get a very good amount of virus particles. Also, I used protein expression with Pichia pastoris to express the capsomeric protein for in vitro self-assembly and disassembly. So these are the results. And these are the results I did with the investigation of self-assembly of particular virus particles. These are the TEM pictures and also the DLSS. So this disassembly and assembly are done with a particular buffer system which is resembles in the cell cytoplasm. For example, how virus is infecting to the cells, any cells. For mammalian cells, so plant cells is different. For mammalian cells, when virus is contact with any cellular mechanism, so initially it will bind to the receptors, surface receptors to the glycoprotein. Then it will engulf inside through the endocytosis. When the virus particles enter to the cytoplasm, so there that buffer will disassemble. Some enzymes will help to disassemble the virus particle and it will release the nucleic acid from that particle. So the nucleic acids will go and find to the hijack the cellular mechanism for their own production. So like that in vitro, I did with some buffer which is resembles of cytoplasmic buffer, physiological condition buffer for the assembly and disassembly of the expressed proteins. So here you can see some encapsulation efficiency of that virus with the nanocargo on magnetic nanoparticles. So there we got some monomeric proteins mixed with the magnetic nanoparticles. All dialysis, the condition will dialysis with buffer of pH like 6. When we did with the pH dialysis in the buffer of pH 6, the protein will disassemble. When the proteins dialysis with the pH buffer 7, the protein will get assembled. So by the time the macronucleic nanoparticle mixed with the monomers will encapsulated with the protein monomer when buffer with dialysis with the pH buffer 7. So there you can see some TEM images showing the magnetic nanoparticles covered with the protein monomers. So here's a graphical representation of full work. So we named as a becomovirus smart nanocargos. So this pan pathogenic virus is a highly plant pathogenic virus named is a top 10 plant pathogenic viruses, which viruses I used for my PhD work. So here we produce a protein monomers, which is assembled in the pH disassemble assembly. So in that particular medium, with the particular pH, we mixed with the fluorouracil, which is anti-cancer compound, and also with the gadolinium ion. So gadolinium ion used for the MRI scanning imaging. So both molecules for it, we mixed with the capsomeres, and the buffer, when we change the buffer to the pH buffer 7, so it will encapsulated with all the fluorosis molecule and gadolinium molecule. So further, the protein, the nanocargos will functionalize with speculation. So speculation means we used PEC molecules for increase the biocompatible properties and pharmacokinetic properties. So here the positive side of the protein nanocargo is the protein is a highly needed molecule for any bioconjugations. So it is a very simple to conjugate any kind of molecules with a simple click chemistry. So I used NHS EDH chemistry for the speculations. So after the speculations, I conjugated with folic acid. So folic acid is a vitamin, is a nutrient for normal cell growth. So there is a beauty is, so cancer cells will need more amount of nutrients in our bloodstream. So folic acid, folic acid will conjugate, and folate conjugate will folate conjugate will target to the folate receptor, which is expressed in the cancer cell. So that modification we did with nanocargos, then we injected into the human mice, sorry, mice. So the nanocargos will go and bind to the cancer cell through the EPR effect. The EPR effect means enhanced permeability and retention effect. It means so when the cancer cell is available, so their surrounding blood vessels will be very big, very large. So whatever nutrients in the bloodstream will, most of them will go and accumulate in the cancer cell. Through the EPR effect, the folate conjugated nanocargos will go and bind to the cancer surface. Through the endocytosis, it will go and the inside of the cancer cell. There, the another beauty is, another positive side of cancer cell is, so inside the cancer cell, the pH will be low, like pH of 6. So in that pH, our nanocargos will disassemble. Our nanocargo will disassemble and release both fluorosil and gadolinium ions. So the fluorosis will go and bind to the nucleus and will do some metabolism inhibitions, and gadolinium ions will remain in the cells. So also in the right side you can see some pictures, mice pictures. There MRI scan picture. There you can see some gadolinium ions is picturized in the mice. So we tried this technique as a trial and error method, but at the end of this technique, we understood that it is very much possible that plant virus nanoparticle can be used for real biomedical applications. So thanks a lot.
Now the session is open for discussion.
Hello. Dr. Raja. Dr. Raja, can you hear me?
Dr. Raja?
Dr. Satya, are you there?
Yeah, he's here, but I'll just call him and see.
Yeah, please.
Hello, sir. Can you hear me? Sir, this is Pratima here.
Oh, hello.
Sir, what is the time limit for the presentation? I'm just.
It's supposed to be 10 minutes, but he has exceeded a lot.
10 minutes. Oh.
Yeah,
Okay.
I think the moderator should give instruction on the time limiting.
Yeah, maintaining the time. Yes, yes.
Yeah, it's very important. Or else, no, we don't have time for all the speakers.
Yes, sir.
We'll stick to 7 minutes presentation and 3 minutes discussion. That would be better.
Yes, sir.
Ma'am, I guess. Pratima, Dr. Pratima.
Yes, sir. That will be better.
Yeah, that's
Yes,
better.
sir, that's better.
Yeah.
So, Dr. Satya, can you please give instruction to the speakers to stick with 7 minutes of presentation so that the rest of the time can be utilized for the discussion?
Yes.
Yeah, sure, sir, sure.
Yeah, thank you.
Okay. Dr. Raja, are you here?
Yeah, yes, ma'am. Yes, ma'am.
Yeah, the judges wants to interact with you.
Yes, ma'am.
Dr. Raja.
Yes, ma'am.
That was a nice presentation.
Yeah, thank you.
And my question is, how do you expect the plant pathogen to infect any mammalian cells? What is the probability that, is it a hypothesis or you have experimented?
Sir, yes. No, yes, sir. There is no clear report still on the date because we tried on a trial and error basis. So initially we tried with the cancer cell line. There we obtained very good results.
Yeah, and results like on what basis that you claim that the results are good?
Sir, the viability of cells. So based on when we conjugated the fluorouracil, so when we used to do with the cancer cell line, based on the cell viability.
No, my question is, if that is the case, then your cells shouldn't be alive. If that is the case.
Yes, sir. Yes, sir. Yes, sir.
But you said it's based on viability. Is quite contradictory. That I sense.
No, viable. No, viability means if it is
I understand
if
that.
cell is viable, yeah. If it is cell is viable, so it is not working. If it is not viable, then if it is killing, then it is working. Based on that, we get some conclusions.
Just based on that, that's a primary study, and you can purely claim that the cell has got infected with the virus and the cancer. I mean, the material that you claim. The
Yes, sir. I understand.
Yes.
I understand.
This is though it is preliminary work. You can't say that the viruses that are exclusive of plant can infect animals because nature is so intricate that they're exclusive. That's why the evolution, it keeps on moving ahead of our intelligence.
So for that, what our lab is deciding in. So because some of my juniors carrying out the work, they are doing with mutation, which some adenovirus protein with expressed with plant viruses.
Okay.
They are doing some experiment with that, sir, for this post.
Yeah, it's fine. But my question is very simple. Like, do you have any other confirmation that animal plant viruses infected mammalian cells or mammalian organism?
No, not also, not also.
No, this is your hypothesis that you are trying to make this plant virus particle as a nanocarrier for any therapeutic.
Yes, sir. Yes, sir. But other thing that plant virus nanoparticle can be used for plant purposes, plant genetic
Yeah,
engineering.
that's fine. I totally agree with you.
Yes, sir.
I
Yes, sir.
completely
Yes.
agree with you. But when it comes to using plant pathogen for mammalian infection for either therapeutic or diagnostics, I don't know how far the results would be. And in my perception, it's bit contradictory. And one more thing, maybe I will suggest you to go for checking with specific rise in antibody towards a specific
Okay.
antigen,
Okay.
so that you can get it confirmed. Apart from cell viability, that is like there could be a number of reasons that cells would die, not just because of this incubation with your viral particle. So best way is to check for any antibody that is raised against a specific antigen. This is one methodology.
Fantastic, sir.
Yeah. Thank you. I'm done here. And how about your one more question, Dr. Raja? And you said you have infected with viral cancer cells. Again, that was cellular, cultured cells, right? That was not in animal models.
Sorry, actually, we are not infected with the cancer cells. Actually, the particle size is very reduced. So we understood.
Raja, your voice is breaking.
Gone with some literatures that through endocytosis, some particle, the size between 10 to 100 nanometer, which will easily go to the endocytosis through the folate receptors.
Yeah,
So
that's
I
fine.
cannot
That's
say,
fine.
I can't say that it is kind of infection. It's kind of endocytosis with.
Okay. All right. And yeah, I go with you. But how about this cancer cell that you said you have made the particle to get into through the animal models? Whether the animal model was the cancerous model or
Cancerous model.
what was the cancer type that was it suppressed or induced
Lung
cancer
cancer, sir.
model?
Induced lung cancer induced.
What was that model? Can you please tell me?
So that work I did with the CLRI. So
Okay.
we just got the
Okay.
results.
Okay. Fine.
So my
So
part is like material kind of things.
okay. So you stop with the preparation of material.
Yes, sir. Yes, sir.
So the final result of this animal infected or as you said, endocytosis. What was the final output that you observed in animal model? Did you assess any?
Yes, sir. Yes, sir. What they told, like they observed that tumor regression.
Now, in that case, you could have shown any specific games test or any other histological analysis that shows the reduction in the cancer size. Like these are my suggestions that would be helpful if at all you are preparing for any further publication of this research.
Sure, sir. Sure, sir.
Thanks, Raja. I'm done here. Thank you.
Thanks so much, sir. Yeah.
Thank you, Mr. Raja, for the informative presentation shared with us. Before moving on to the next participant, We kindly request the participants to please stick on to the time limit, which is 7 minutes for presentation and 3 minutes for the question session. Moving on, I now welcome Kausalya to share the views on the topic evaluation of primary and secondary metabolites of Artabotrys hexapetalis.
Ma'am, whether my screen is visible, ma'am?
Yes, it's visible.
Okay, ma'am. Okay, ma'am. Thank you. Happy noon, all. I am Kausalya, research scholar, Department of Biochemistry, PhD College of Arts and Science. I am going to present my research work on evaluation of primary and secondary metabolites of Artabotrys hexapetalis bandari leaves. I have done this work under the guidance of Dr. D. Victor Arogedas, Associate Professor, Department of Biochemistry, PhD College of Arts and Science. Introduction. Plants contain natural bioactive compounds called as phytochemicals. Generally, this phytochemical is classified into two types. As we know, primary metabolite and secondary metabolite. Carbohydrates, amino acids, chlorophyll, proteins are primary metabolites. Secondary metabolites include alkaloids, flavonoids, terpenoids, tannins, phenols, glycosides, and saponins. Medicinal properties of a plant is due to secondary metabolites. Quantification of primary and secondary metabolites will be helpful to identify bioactive compounds. These bioactive compounds have therapeutic value in treating disease. The main aim and objective of the study is to assess the phytochemicals of Artabotrys hexapetalis bandari leaves using different solvents like water, ethanol, acetone, chloroform, and petroleum ether, and to evaluate the primary and secondary metabolites of aqueous leaf extract of Artabotrys hexapetalis bandari. So this is the plant which I have selected for my study, Artabotrys hexapetalis leaves. This plant was identified and authenticated by the senior scientist, Botanical Survey of India, Southern Region Center, Tamil Nadu Agri University. It belongs to the family Anonaceae, genus Artabotrys, species A. hexapetalis, binomial name Artabotrys hexapetalis bandari, common name it is called as Manoranjidam, and the major part used is leaves. Coming on to the methodology, preparation of plant extract. The leaves of A. hexapetalis were shaved, dried, and ground to coarse powder. The dry powder, that is 20 grams, was extracted using different solvents like water, ethanol, acetone, chloroform, and petroleum ether in the ratio of 1:10. And this is used for qualitative analysis of phytochemicals. And for large scale aqueous extract preparation, 60 grams of plant powder was soaked with 600 ml of water. So at last, 8 grams brown fine crystals were obtained per 60 grams of plant powders. And this was stored in airtight desiccators for various assays. So this is the general methodology which I have followed. Alkaloids, flavonoids, saponins, carbohydrates, amino acids and proteins, phenols, glycosides, tannins, sterols, and terpenols. This is the basic phytochemical which was followed. And the estimations of total carbohydrate was done according to Hedge and Hofreiter, and the estimation of protein was done according to Lowry et al. Method. Estimation of free amino acid was done according to Muir and Stein. Estimation of flavonoids, estimation of tannins, and estimation of total phenol was done according to the standard procedures. Coming on to the results and discussion. So phytochemical analysis of Artabotrys hexapetalis leaves revealed that water extracts shows the maximum phytochemical activity. So when compared to 5 different solvents like water, ethanol, acetone, chloroform, and petroleum ether, water extracts, that is aqueous extracts, reveal the presence of alkaloids, flavonoids, saponins, carbohydrates, proteins, phenols, glycosides. So this was taken for further estimations. Tannins and sterols was also reported in the aqueous extracts of A. hexapetalis leaves. And quantitative analysis of plant constituent was done. So in the protein concentration was found to be 60.4 milligram per gram of leaf extract. Carbohydrate concentration was found to be 43.16 milligram per gram of leaf extract. Amino acid concentration was found to be 19.33 milligram per gram extract, and the flavonoids was reported to be 28.3 milligram per gram extract. Tannin concentration was found to be 24.53 milligram per gram extract. At last, the phenol was found to be 7.63 milligram per gram extract. Coming on to the conclusion, aqueous extract of A. hexapetalus leaves showed the presence of alkaloids, flavonoids, saponins, carbohydrates, proteins, amino acids, phenols, glycosides, tannins, and glyterpenes. The present study revealed that A. hexapetalus leaves are a good source of secondary metabolites, which can act as a natural antioxidant. And future perspectives, isolation and characterization of bioactive compounds from A. hexapetalus leaves. This is a reference follow. Thank you. Thank you for giving me this opportunity to present in this conference. I would like to thank the staff members, Department of Biotechnology, for organizing such a wonderful virtual conference. Thank you, ma'am. Thank you for giving me this opportunity. Thank you all. Ma'am, I have completed my presentation, ma'am.
Yeah, now over
Yeah,
to
we
the
have noticed.
judges.
We have noticed. Yeah, we have noticed. Kausalya.
Kausalya, I have a question.
Yes, ma'am.
What is the reason behind selecting the leaves of this plant? The flowers are very fragrant, usually we use it for, even we wear it on the hair, it is very fragrant and it has a very mixed fragrance. So why you didn't try anything with the flowers? What is the reason behind selecting the leaves?
Ma'am, flowers quantity is very difficult to collect, ma'am. So I have gone with leaves, ma'am. And leaves contain so many bioactive compounds, ma'am. So I have selected this for my study, ma'am. When compared to flowers, leaves consist of so many antioxidant activity and bioactive compounds, some of the peculiar compounds, ma'am. So I have taken this leaf for my study, ma'am.
Okay, like you have used only like 60 to 40 to 60 grams of dried powder, right?
Yes, ma'am.
Flowers, I think that much of flowers you will be getting. Even in the market, you can just get for the study sake.
Yes, ma'am. But the bioactive compound which I am targeting for my animal study is highly present in leaves, ma'am. So I have selected for this leaves for my study, ma'am.
So this is a very preliminary work that you have just begun.
Yes, ma'am. I have started, ma'am.
Okay. Thank you.
Thank you, ma'am. Thank you, ma'am.
Thank you, Kausalya. I'm sure of the fact that you.
Kausalya, one more question I have.
Sir?
Can you hear me?
Yes, sir. Yes, sir. I can hear you, sir.
Yeah, actually, the title says that it's evaluation of primary and secondary metabolites. But
yes,
you spoke mostly about the identification.
Just I have started with the work, sir, phytochemicals, and I have quantified carbohydrates, so and so. Just I have started.
In that case, you could have changed the title into so. I was expecting, evaluation in the sense like, what is the impact of or recognized or so identified metabolites and so and so. Yeah, that's what my expectation.
Okay, sir. Okay, sir. Sure, sir.
How do you expect this plant derived secondary metabolites would make an impact on public life or scientific community?
Yeah, even I had the doubt, sir. Like, what is the common use of this plant other than the fragrant flowers? Is there any other medicinal properties for the?
Yes, ma'am. It is used in traditional medicine to treat diarrhea, even in kidney diseases, so and so, ma'am.
Okay.
In folk medicines, it is used, ma'am. And the flowers, due to its fragrance, it is used in aromatherapy. And not much research is done in this area, but this leaf is reported to have high bioactive compounds, ma'am. So there is an evidence for that. So I have selected this plant, ma'am.
Yeah, that's what I wanted to.
It will be helpful to treat this pandemic condition, ma'am. Maybe the vitamin C concentration, vitamin E, if it is high in this leaf, it is reported now, so that it will be helpful to even cure this COVID. Helps to boost the immune system, ma'am.
Like normally, I haven't heard of using these leaves for any medicinal property. So I was very curious to know what it actually does.
I mean, in Chinese medicine and in folk medicine, tradition medicine, this leaves and flowers is used to, ma'am, to treat
Okay.
diarrhea, kidney diseases.
Okay.
In some area, it is used to control even fertility in rats, ma'am.
Mhm.
Fertility of rats, ma'am.
Okay. Thank you.
Thank you, ma'am. Thank you, ma'am.
I hope there are no more questions. So moving on, I now welcome Ms. Niranjana R to enlighten us on the concept synthesis and characterization of chitosan-based hydrogel and its in vitro antibacterial activity.
Good afternoon. Is my slide visible?
Yes, ma'am.
It's visible. You can go ahead.
Good afternoon to one and all present. I am Niranjana. I am currently pursuing my MSc in Biotechnology at PSD College of Arts and Science. My topic for today is synthesis and characterization of chitosan-based hydrogel and its in vitro antibacterial activity. So, hydrogel can be defined as a 3D network of hydrophilic polymer that can swell in water and hold large amount of water. At the same time, it does not lose its structure because of chemical and physical cross-linking. They have the following characteristics, like they are biodegradable, biocompatible, non-toxic. Hence, they are generally used as they're generally used in drug delivery systems. Currently, there are research going on regarding their use as for wound healing property. So, chitosan-based hydrogels that have high antimicrobial agents can be used for wound healing because they provide a moist wound environment and they help in reepithelialization. So the objective of the study is synthesis of chitosan-based hydrogel, evaluation in vitro antibacterial activity, and evaluation of heme agglutination activity. The chitosan is derived from fish scales. It is also to the hydrogel potato peel powder and orange peel powder is also added because they are found to have antimicrobial properties. So for the extraction of chitin and chitosan powder, the fish scales were first washed with clean water and dried. It was then soaked in 1% HCl solution for 36 hours. It was then washed and soaked in 2N NaOH for 36 hours for demineralization. Further, it was treated with potassium permanganate for decolorization. This leads to the production of chitin. From the chitin so obtained, it is again soaked in 50% NaOH. This leads to deacetylization and results in chitosan. So from the chitosan so produced, hydrogel is formulated. 1 gram of prepared chitosan was weighed and suspended in 20 ml of 2% acetic acid at room temperature for 24 hours, and it is constantly stirred. This leads to a pale yellow gelatinous chitosan solution. To this solution, we add potato peel powder and orange peel powder. And it is again continuously stirred for 5 hours to obtain a viscous brown chitosan potato peel powder and orange peel powder solution. To this solution, 1 ml of aqueous glutaraldehyde is added and it is stirred for 5 to 10 minutes. This aids in cross linking of the chitosan hydrogen.
So this is the chitosan-based hydrogel that was obtained. Moving into the results and discussion. So the SEM images are of the chitosan and chitin and chitosan from the fish scales. Both of them had a rough and thick morphology. This is the SEM image of the chitosan-based hydrogel. Both of them showed expansion and they had a stiff morphology. So we tested the antibacterial activities through agar-well diffusion method. And the gray bar in the graphical representation shows the efficacy of chitosan-based hydrogel. The efficacy of chitosan-based hydrogel was tested against E. coli. It was tested against E. coli, Staphylococcus aureus, Pseudomonas, and Klebsiella species. So this is the agar-well diffusion that was performed. Heme agglutination assay was also performed, and it was found that the hydrogel did not cause any agglutination of blood cells, and there was no clustering or any of those things was not observed. Moving on to summary and conclusion, 7 photographs exhibited expansion and stiffness surface morphology. The antibacterial activity obtained was good. Further, we would like to increase the standardization and allow more studies. We would also go in for we would also like to go in for animal model studies, etc. Thank you, sir.
Hello, Ravindra.
Yes, sir.
Nice presentation.
Thank
it was.
you.
And, why did you choose fish scale as a base for the hydrogel crystal?
The exoskeleton of a number of organisms like crab, fish, they contain chitin. Extraction of chitosan from these sources is what we intended to do.
All right. And how our hydrogen-based hydrogen is different from the usual hydrogen? What tests you need advantages over the other materials?
Chitosan is known to have a good biocompatibility, so that wouldn't be an issue. So that is why we tried with chitin.
Like you can number of other materials are there already, like PLA or PLGA, like poly PLGA or so. Like either they do have compatibility. What's
thing,
new in this is the question.
The thing is, fish scales are easily available and they're generally a part of the fish that is not really used. So this could be a way of using something that's generally neglected, which is not used.
But there are more chances that it would get recognized as a foreign material, obviously.
Okay, sir.
When it comes to therapeutic purpose, so that if you are tagging any drug along with this, or if you use this material as a scaffold for the tissue engineering, don't you think that grafting is possible?
Sir, one of the reasons why we went for the heme agglutination assay as a preliminary assay was to see whether it had any toxic effect. But then further, if we have to see if it is incompatible, then we'll have to go for further analysis, which we've not done as of now.
Does the heme agglutination results may not be much promising one to control that.
Right, sir.
Right. Do you claim any other advantages or other materials?
Sir, as of now, we've just looked into it in the perspective of it having antibacterial property such that it can aid wound healing. So, in case of wound healing, it could be a better option than other wound dressings because other wound dressings they could injure, like most of the wound dressings they cause irritation to the wound site, whereas hydrogel they provide a moist environment for the wound, so the wound will heal quicker.
So your intended purpose is either towards mammalian or microbes? It's my another question. Is it
So wound healing for a mammalian?
okay? But you said antimicrobial.
So wound generally wounds have a potential of getting infected by other microbial agents like S. aureus or other microbes. So a hydrogel hydrogel containing antimicrobial properties could aid in wound healing. That is what I intended.
Okay. Do you have any idea to conjugate any material along with your hydrogel, or just you're using a plain hydrogel that?
The hydrogel that we've worked right now, we've incorporated orange peel powder and potato peel powder because both of these have shown results that they have antimicrobial properties.
So how's the stability of this material as a scaffold?
Sorry, sir, I couldn't hear you.
What is the stability like? How long or how promising this material as a scaffold because of tissue engineering?
So we haven't studied it as a scaffold. We've only studied its in vitro antibacterial property.
Okay, but we have future perspective.
Yes, the future perspective includes probably studying it as a scaffold.
Right. Thank you.
Thank you, sir.
I am done here. Over to you, Dr. Pratima.
Yes, sir. Niranjana, how do you store this hydrogels, prepared hydrogels, and what is the, keeping conditions for this hydrogel? Like how long can we store it? The shelf life, because it is a polysaccharide, it might also be prone to microbial degradation. So, is there any information with you?
Ma'am, I think she left the meeting.
Okay.
So to answer this, actually, I was also involved in that project. So excluding that, I will just answer this part. We didn't see the stability, but it was stable for 5-6 months. 5.5 months in room temperature. So after that, it was not stable. So we are just working on it to stabilize in aspects of stabilization, and also this is the juvenile stage, so we are working on it.
Nice work, Satya. And also, I wanted to know whether is there any eco-friendly way for deriving this chitosan compound from these waste from the fish. this fish
Yeah,
and
yes. There are so many
using
methods.
more amount of acid and salts are used for deriving the compound, right? So is there any methodology that can be modified for deriving this? That would be really good work.
Yeah, ma'am. That will be promising. So as of now, we use the very preliminary that acid treatment. So this was the first work we initiated. As you know, I was working in plant system and I have
Yeah,
just
yeah.
moved towards it. So this is the juvenile state. So right now, I just follow. I mean, we just follow that preliminary step. So we are working on it. Maybe we will find some eco-friendly ways sooner with less use of acid, and also we need to get more amount of the product like the chitosan. So we are working on that in almost all the crustaceans.
Okay, Satya. All the best.
Thank you, ma'am.
Yeah, thank you.
Thank you for another informative presentation. I once again like to kindly remind the participants to please stick on to the given time. So now moving on, I now warmly welcome Manju Vidya Shakti Kumar to share the contents on the topic reductive stress, an emerging concept in the cardiac hypertrophy.
Good evening, everyone. Am I audible and is my screen visible?
Yes, Manju.
Okay, thank you, ma'am. I'll start now. The topic that I've chosen for today is the reductive stress and emerging concept in cardiac hypertrophy. I'm Manju Vidya Shakti Kumar, doing MPhil under the guidance of Dr. Victor Arogedas from the Department of Biochemistry, PSG College of Arts and Science. Cardiac hypertrophy is nothing but the abnormal thickening of the heart muscle, which occurs in response to intrinsic or extrinsic stress, which happens during many conditions like continuous exercise, diabetes mellitus, and it causes modifications in the cardiomyocytes and causes remodeling of the heart, which leads to congestive heart failure, myocardial infarction, and sudden death. Reductive stress is a counterpart of oxidative stress, in which, in this case, shifts the redox homeostasis of many important biological redox couples like reduced and oxidized glutathione, NADP and NADPH, NAD, NADH to a reduced state. What happens is when there is an excessive antioxidant generation through the overexpression of NRF2, and when the NRF2 is overexpressed, antioxidant enzyme is also overexpressed, and it increases the levels of reducing equivalence, which causes damage in the heart and other tissue damages. The plant that I've chosen for my study for the treatment condition is Brassica oldacea, which is broccoli. It contains a major phytochemical component called sulforaphane, which is present more in the broccoli, which is known to regulate the NRF2 expression. When the hypothesis of the study, in the animal models, I'm injecting isoproterenol to induce cardiac hypertrophy. When the isoproterenol is used for injection, it hyperstimulates the beta adrenergic receptor, which causes a shift in the energy substrate utilization. Normally in heart, the glucose and free fatty acids are the major energy substrate, but in this case, glucose and lactose will be utilized more, which leads to increased ROS generation and normally leads to oxidative stress. But when there is a chronic activation or upregulation of NRF2, it leads to reductive stress condition and causes changes in the cell signaling pathways like MAPK and other antioxidant defense pathways, which leads to the pathological remodeling, contractile and mitochondrial dysfunction and fibrosis, and ultimately leads to the development of cardiac hypertrophy. When the plant extract of broccoli is given to the rats, it causes it causes changes in the major pathways, like as I mentioned, like MAPK, PI3K, and PKC, and causes changes in the NRF, causes modifications in the NRF2. NRF2 is usually associated with keep1 complex, but when it gets phosphorylated, it binds to it goes inside the nucleus and binds to JUN, and it binds to the DNA, which will cause the expression of antioxidants enzymes like hemoxygenase 1, NQO, and glutathione peroxidase, and this regulates the NRF2 expression. This will prevent cardiac hypertrophy. Objective. The study is carried out with the objective to understand the relationship between reductive stress and cardiac hypertrophy, to assess the reductive stress by measuring the ratio of oxidized and reduced glutathione, NAD plus and NADH, and analyze the activities of enzymes like glutathione peroxidase and glutathione reductase, and to evaluate the efficacy of the plant extract and the amelioration of cardiac hypertrophy. The experimental models, I've taken 4 groups of 6 animals each. We have taken male adult Wistar rats. First group is normal, which is given saline, and the second group is treated with isoproterenol and the dosage of 10 milligram per kilogram body weight, 7 days. Isoproterenol is injected intraperitoneally. And the third group, isoproterenol is injected, and it is simultaneously treated with the standard drug losartan, which is given the dosage of 50 milligram per kilogram body weight orally. And in the fourth group, isoproterenol is injected and treated simultaneously with the aqueous extract of broccoli in a dosage of 300 milligram per kilogram body weight, 7 days orally. At the end of the study, rats were dissected and the blood is collected through cardiac puncture, and the heart is dissected, and the tissue homogenate is used for biochemical analysis. The reducing equivalents like reduced glutathione and GS, and the value of GSH and NADH is measured. And antioxidant enzymes is also assessed, like glutathione peroxidase and glutathione reductase. Coming to the result and discussion, these are the reduced glutathione and GSH and GSSG ratio. As you can see in the second group, that is the disease-induced group, there is a significant increase in the levels of reduced glutathione and GSH, but in the treatment group, they are brought back to normal. And as well as in the case of glutathione peroxidase and glutathione reductase, that is the same case. And the induced group, it is significantly increased, but those values are brought back to normal in this thing. And the glutathione reductase and glutathione peroxidase is the important enzyme which is involved in the redox homeostasis of glutathione, and it causes, when it is produced in more, it causes the H2O, hydrogen peroxide, to be generated more, and it causes tissue damage. And these are the NAD plus and NADH ratio. It is decreased in isoproterenol induced group, which means that the level of NADH is increased in the disease induced group, and it is brought back to normal in the treatment groups with the standard drug and plant extract. And conclusion, these results show that there is an increase in reducing equivalence. Until now, the focus is mainly on the oxidative stress, but these results indicate that reductive stress also plays a key role in the development of cardiac hypertrophy. And nowadays, the antioxidant-based therapeutic approaches are growing more, so we should take a proper evaluation of the redox state before the treatment. Future perspective, the molecular mechanism between the reductive stress and cardiac hypertrophy is still unclear, and hence further studies should be proceeded on that. And the role of other signaling pathways related to cardiac hypertrophy should be studied for identification of novel targets. These are the references which I have used. Thank you for your time. Hello, ma'am.
Manju, I just have a very layman question.
Okay,
So eating this broccoli or any other cruciferous vegetable will help in reducing the stress?
Ma'am, all the cruciferous vegetables are known to have this sulforaphane and other isothiamate compounds, but sulforaphane is present majorly in broccoli.
Okay, like the content is higher than
as other vegetables. Compared to other vegetables, broccoli has especially broccoli, the seeds have high content of sulforaphane.
Okay, broccoli seeds, not the broccoli florets.
Ma'am, florets and as well as the seeds, ma'am, both of them has a high content of sulforaphane.
Mm-hmm. Okay, thank you.
Manju?
Yes, sir. Hello, sir.
Hi, can you hear me?
Yes, sir, I can hear you.
Actually, it's well-known fact that broccoli is rich in antioxidants.
Yes, sir.
And sulforaphane. It's a known fact that.
Yes, sir.
It's known one. That's why people
No,
prefer.
sir. Actually, broccoli is mostly studied for oxidative stress, sir, but in this case, actually it is known to increase the value of antioxidants. But in this case, I'm studying the reductive stress, sir, like how it is performing in the reductive stress and for cardiac hypertrophy conditions, sir, because broccoli has other compounds too.
That's what I wanted to mean.
Okay.
What are the other things that specifically contribute towards, as you said, reductive stress?
Yes, sir. Sir, I can't hear the last part of the question properly, sir.
What are the? You said that there are other parts.
Other compounds like major
Components, yeah.
Yes, sir.
That contribute to reductive stress that you mentioned.
Yes, sir. Yes, sir. So usually broccoli is known to increase the level of antioxidants, but in this case I'm studying like how it is performing in the condition of in the case of reductive stress, like whether it is whether it can regulate the expression of NRF2.
Yeah, but studies you have shown it's merely biochemicals, right?
Yes, sir. So this is an ongoing study, sir. This is this is only part of my work, sir.
So it's still in the hypothesis level. It's not at
no, sir.
Like how you're going to analyze the further parts of your research?
So we are going to do the molecular level and other this thing also, so metabolic, how it is performing in the metabolical enzymes markers like G6PD and how it is affecting the other markers of cardiac hypertrophy like LDH, CKMB, other things and all also, sir. Reductive stress and like antioxidant is just a part of my work, sir. I'm also working on how it is performing in glycolysis, glucose metabolism, other this thing also, sir.
When it comes to cardiac hypertrophy,
yes, sir.
How it's going to work on that? Like, do you have any plans to check directly in the model? They have, apart from including through tertiary and secondary analysis.
Sir, we are also monitoring the ECGs of the rats during the study.
That's what I mean.
Yes, sir. We have performed ECG, sir, for the rats, and we have got good results with that, sir.
Maybe you can check the ultrastructural change.
I can't. What, sir?
You can check. Maybe you can check with the ultrastructural change.
Sir, we are also giving for histopathology and also the other changes also, sir.
Yeah, fine. Thank you much.
Okay, so thank you.
Thank you for another fact-based presentation. Participants, please beware of the timing. And now I call upon Dharini Ambiguri, Department of Biochemistry PhD guest, to share the contents on understanding the relationship and identification of predictors for cardiac hypertrophy using correlation and regression.
Good evening, everyone. Am I audible? Hello?
Yes, you are audible.
Yes, ma'am. Yeah, thank you, ma'am. And is my screen visible too?
Yes, it's visible, but you can go for that slide share.
Okay. Yes. So good evening, everyone. I'm here to present the study with the entitled topic, Understanding the Relationship and Identification of Predictors for Cardiac Hypertrophy Using Correlation and Regression. And this we have done it as a pilot study. And I'm doing a PhD under the guidance of Dr. Victor Arogidas, the Department of Biochemistry. So coming to the introduction, actually speaking, as biologists and as researchers, we publish a lot of data and a lot of research articles and everything, and we're aware we have n number of data with us. But just showing like we induce a disease to a rat and we treat it, or we do some researches like that and we end up showing it is getting treated. We think yes, we are showing some result, but is it so? If we think actually we are, I would say it is completely sort of incomplete task, because nowadays researchers have becoming interested to take up the already available data in the data repositories as well as ongoing researches, and they are trying to bring they're trying to understand relationship between parameters using various mathematical models and computational models, which is termed by the name of machine learning algorithms, and most probably would have heard nowadays like big data analysis, all these things have the term called machine learning algorithms, which they use to retrieve, some information out of the available data. So here I have shown an example of COVID-19, which we are familiar with, because in COVID we are aware that n number of researches have happened across the world. It is like as shown here, epidemiological data we have clinical data, we have genetic data. People have shown this gene gets expressed or this gene gets suppressed during treatment, and this metabolic parameters gets changed. So everyone have each data with them. But as a collective information, what can we do with that is what nowadays people are trying to focus on. So what they do is they try to use machine algorithms like correlation, regression. These are some basic fundamental items, and they try to find out relationship between those parameters, so that which they can use. Like for example, if a COVID patient from India versus COVID patient from Africa, and one patient is having high blood pressure and another patient is having diabetes. So how for which gene is getting switched on, which gene is getting switched off. So those correlations they can study, and people are trying to find out relationship between individual parameters nowadays. So what is the benefit out of it is this can lead to preventive medicine, new focus on therapeutics, new diagnosis operations and all they can do with the help of such machine learning algorithms, because they train the data sets using the available information which is already dumped in the data sets. So in our study, what we did was we focused on one particular disease, that is cardiac hypertrophy, as spoke by the previous speaker, Manju. Cardiac hypertrophy is a disease where you can see the left ventricle is getting inflamed. So the causes may be because of diabetes, hypertension, chronic kidney diseases, and even cancer patients who take anti-cancer drugs are prone to this clinical condition. So what we thought was like, okay, we will take a basic clinical parameter, which is serum glucose. Glucose is a culprit in most of the diseases, and just like that in cardiac hypertrophy. And albumin, serum albumin is one of the biomarker in cardiac hypertrophy wherein it gets reduced in hypertrophic conditions. So we took those two clinical parameters, and what we did was we used two diagnostic parameters, that is ECG's peaks. In the ECG, we know there are different peaks, and among them the QRS peak, the complex, as well as the RR interval, that is the space, that is the gap between the one beat to the next beat, how far the heart is beating, good or bad, that is RR interval. So we took those two. So totally 4 parameters we took, and we thought, okay, we'll study the relationship between them and how far it influences each other was the objective of our pilot study. And coming to the methodology, what we did was we took this data from our own published previous article itself, because just like the previous speaker, the questionnaire session, as sir was telling, we did some ECG data for rats also. So we took those ECG data, QRS complex, RR values from our published data, as well as the same where we got the levels of glucose, albumin. We took all those 4 from our own data, and we tried to do a correlation and regression analysis. And for correlation, we used an online software called Metabo Analyst. It is an open free online tool, and followed by which we try to perform multiple regression analysis using our familiar SPSS software. So coming to the results, this is the correlation heat map, I would say. Just don't get bugged with the colorful thing. The point here is that you can see the diagonal one. If you see from the left top to the right bottom, it will be completely red, which means it is just a matrix format. So wherever you can find the complete dark, it is positively highly correlated.
Excuse
Yes,
me.
ma'am. Yes, ma'am.
Can you put in your slideshow format because the slides are not moving. It is still in the
Is
first
it a
slide.
problem for everyone, ma'am?
I'm not sure.
I too sense the same. You are still in the very first
First
title slide.
slide. Yeah. We did not see the second or the third slide. It is still in the first.
Yeah.
Now, can you
Now
put
it's
it
okay.
on the slideshow? You see in the right corner.
Yes,
Yeah.
I had actually put up in the slideshow only, ma'am.
Okay, we did
Actually,
not.
I was and now is it okay?
No, but it is okay. At least the slides can be moved.
Okay, but actually it's in the slide mode. I'm so sorry. I don't know what happened, ma'am. Should I give another brief introduction or should I move on, ma'am?
Please move on. You were explaining the heat map, right? You can go to that slide.
Okay. So regarding this, if you could see, it is just a matrix format of idea. So if you could see compare the serum albumin with the serum albumin, definitely it is the same. It will give a dark red color because it is a value of 1. So the same way you just correlate two different every conditions, and if you see, you can very well see the serum albumin will have a negative correlation, whereas the serum glucose will have a positive correlation, which can be better viewed in this pattern search format. You can definitely see here the correlation coefficient values of serum albumin is negative, whereas that of the serum glucose is positive with the ECG data, which means we can see that okay, if the serum albumin is getting reduced, then the ECG values will be increasing. So it is inversely proportional. So it is not the case with glucose. If glucose is getting increased, then definitely there will be a subsequent increase even in the cardiac events also. So that is one of the things we could get an idea. So we thought, okay, actually initially we thought, okay, serum albumin is only showing negative. So maybe that is only most prominent one we thought. So when we did regression analysis in SPSS, we got data like, please do not get panicked about the numbers. The simple concept here is about the circled one. You can see at the bottom, the standard coefficient beta value for serum glucose is higher than that of the serum albumin. Yes, ma'am.
Slide is not moving. It's in introduction slide.
Is it moving now, ma'am?
Yes, ma'am. It is moving.
Okay,
now only it is moving.
Okay, fine. So here you can see the beta value of the serum glucose is high when compared to the serum albumin value, which means like serum glucose is having more impact upon the QRS events. That QRS means it is the ventricular depolarization activity of the heart. So it is having an impact upon that, and we can withdraw the equation of this is a predictor equation. So what we can do with the equation is we can substitute the concentration of glucose in this X value. Maybe if you diagnose a patient with a glucose value of 200, just substitute the value of 200 here, you will get to know the QRS value of that particular patient, and you can get an idea whether he may be prone to hypertrophy in later stages, or is he already in a hypertrophic state like that. So this is the ability of using regression. We can predict and we can build up prediction models using regression for our clinical conditions. The same way with that of the RR interval prediction also. Even there we found like serum glucose is having better impact when compared to the serum albumin, and we obtained a prediction equation. So conclusion is this is actually only a pilot study. As I said, it's a very fundamental study, but the main idea is we want to bring out like we should never stop at telling like, yes, this causes diseases, this treats it. But in between whatever parameters we take, we had to, nowadays we have, the need is to bring out the relationship between individual parameters so that we can get an idea of like whether we would be, it will help us to predict the early diagnosis of the diseases, not only for glucose, you can use it for even metabolomic analysis. And nowadays, a lot of metabolomic and proteomics people are using this metaboanalyst to get some such useful insights from the data. So I would say it is a take-home message like we have to implement such machine learning approaches even in our biological researches so that we can come up with some new diagnostic insights and preventive options. So these are the various key references. Thank you. Hello? Hello, ma'am?
Yes, sir. You're audible, Dharani Ambika. Over to the judges.
Okay.
Dharani?
Yes, sir.
Yeah, can you please scroll down your slides so that I could see it once? I didn't see your slides.
Okay.
Yeah.
So this was just an introduction, sir, about the clinical condition and the need for using machine learning algorithm. And this was the objective. What were the 4 parameters we chose? This was the methodology. Actually, we took our own published data to perform this pilot analysis. And these are the results, sir, actually. The Pearson's heat map, and it is better viewed with the pattern search analysis, followed by multiple regression analysis. So this was for RR and the conclusions.
So you'd like to conclude the parameters, the endpoints,
Yes.
and the parameters by using regression and correlation.
Yes, I would like to say adding correlation and regression to every researchers might give further insights and a relationship or associationship between our own parameters, whichever we study. We can even narrow down. Sometimes cholesterol may have only weak effect upon our hypertrophy or any disease we study, but we might have been focusing on it much more. So I think such analysis will definitely help to further narrow down or pinpoint or give better clarity for our researchers.
Okay, did you check with any other specific data?
Yes, actually, yes, sir. We actually developed 7 models of cardiac hypertrophy, and we have done GCMS analysis, and we have done a metabolomic analysis, and this using this metaboanalyst, and we have narrowed down different predictors. Actually, it is under submissions for journals, so that is why I'm unable to show the results. So sorry. But we have performed GCMS analysis. Yes, we can do it, sir. We have done it also.
So you have focused on cardiac hypertrophy.
Yes, sir. So this can be applicable even for any other diseases. It is just an idea which I'm trying to project on.
Okay. So it's under consideration for a publication or what?
Yes, sir. One is already both among the 7 papers. We have submitted only 2 because the remaining 5 I'm just building up the manuscript. Both of the thing are under peer review, sir.
All right. How many research articles you have considered for a single?
Sorry, sir. I've submitted both. Both are under review, sir. The remaining 5 models of hypertrophy, I'm just now building up the manuscript. So I've submitted 2, and both are under review, sir. There's not a
My question is,
yes.
How many research articles you considered for your?
This is a pilot study. I started this only with our own published article because I thought in case I have to use someone else's.
Is it enough to conclude?
No, that is why I put because the reason why I thought that would be sufficient was in that study we took a minimum of around 6 animals per group in that particular paper. So whatever values would be statistical based, and that's why I chose only that paper. But I would say yes, definitely, as you say, we need more papers. Maybe we need to get the permissions, I guess, right? So maybe in case we are using any other people's data for meta-analysis or something. So that's why I did not use other papers. I initially started, this was done initially at initial stages
because if you consider your own publication,
yes,
output would be very limited, and
yes, exactly.
That might not give accuracy for your conclusion. That's what I mean.
Okay, so fine. Yes. So this is just a idea that I would like to project it because a pilot study could give this much. I thought maybe, as you said, if you could do more, that would give more insights. As you said,
it's a sort of meta-analysis.
Yes, sir.
That's nice.
Yeah, thank you, sir. Thank you.
Over to you, Dr.
Yes, sir. I don't have any questions for this because it's not my area. But it's a general question. Can you develop any algorithm so that you feed these data so that you
no,
can...
to be honest, I am not good at developing algorithms, ma'am. But my guide, Dr. Victor, he does it actually. He develops algorithms and the coding, everything, and all for that. He's good at it. To be honest, I'm not good at developing algorithms, ma'am. I just use the available algorithms and programs to do analysis and get the output out of it. That's it, ma'am.
Because regression and correlation are very basic statistical studies used for any kind of data.
Yes, yeah, yes.
You have this glucose and this cholesterol.
Yeah, that's me. Exactly. Yes, we can even use. Usually, people use, as you said, they use microarray data. Everything they use for yes. But as this is a pilot study and initially we wanted to try with the basic things that affect hypertrophy, we chose glucose and albumin, ma'am. That was the reason. Yes, ma'am.
Okay. So are there any modeling software like that?
Yes, yes, that's what I said. A metabo analyst is one such things which we felt is a feasible one. There are other softwares also available online, ma'am. Yes.
Do we have anything in R? The R is very...
yes, ma'am. We actually metabo analyst, you can use it using R coding also. It gives two options for you. You can use a coding version as well as you can use it in a Windows platform also. I mean, it's like this one.
Okay, thank you, Dharni.
Thank you.
Thank you for the presentation. I warmly welcome Mary Joy, Department of Biochemistry, PhD CAS, to share the contents on the topic Beneficial Effect of Plant Extract Cisbania bispinosa on enzymatic and non-enzymatic antioxidants in depression-induced animal models.
My voice? Can you hear my voice? Anyone?
Yeah, you're audible. And go ahead.
Yeah, okay. Is the slide is visible?
Yeah, but it's not proper. Oh, it's okay.
It's visible. It's visible, Mary Joy.
Yeah, okay, ma'am. Thank you. It's Mary Joy. I'm the PhD scholar of PhD.
Your voice, just a moment, Mary Joy, your voice has got so many disturbances. Your voice is not clear and you have background noise.
Okay, ma'am. Just a minute. Just a minute, ma'am. Is my voice is clear now?
Yeah, it's better.
Yeah, okay, ma'am. Good evening to everyone. This is Mary Joy, PhD scholar, PhD College of Arts and Science. I'm doing my PhD under the guidance of Dr. D. Victor Das, Associate Professor, Department of Biochemistry, PhD College of Arts and Science. Here I got going to share my topic, beneficial effect of plant extract Cisbania pipinosa on enzymatic and non-enzymatic antioxidant in depression-induced animal model. Depression, it is a common psychotic illness that affects mood, thought, physical health and behaviors of any individuals. According to the WHO Department of Mental Health, depression is a significant contributor to the global burden of disease and affects people in all the communities across the world. Indian medicinal plants and their varieties have an invaluable source of therapeutic agent to various disorder for treatment, even including a depression. So here I have chosen the plant called Sesania pipinosa for reducing stress, impairment, and ameliorate depressive symptoms. This Sesania pipinosa has been comes under the family of Fabaceae, and the genus is Sespania. The species is Sespania pipinosa. The Tamil name for my plant is Sittagati. The common name is Sespania p. Aim and objective of my study is I'm going to give oxidative stress by the immobilization stress, which was measured in the term of free radical scavenging enzyme activities. The antioxidant potential of Sesania pipinosa lives and its active constituents were also studied on pre and post immobilization stress induced oxidant and pro-oxidant state of rats. For my study, I have taken the animals, that is Mr. albino rats. My experimental setup is I have divided the animals in the 4 groups. Each group carries 6 animals. Totally 24 rats will be present in my experiment. In group 1, it has been belongs to control group, and group 2 belongs to depression induced. The induction of depression will be taken out by the Crohn's method. Group 3 is belong to the drug treatment. Here I am given the standard drug called imipramine. Belongs to plant extract treated. That is Sesania pipinosa plant has been given to that group 4. Depression was induced by CMS protocol in albino rats. This protocol is given in this slide. I have given food deprivation for 24 hours, water deprivation for 12 hours, solid bedding for 22 hours, cage tilting for 24 hours, and I put the animals in the crowded housing in 12 hours, and the animals are exposed to the novel odor for 12 hours. The cold stress has been given for 20 minutes. Light elimination was given for 2 to 4 days. And finally, the animals were kept in the rotary shaker for 10 minutes. After I'm giving this Coombs protocol to the albino rat, the status of depression was diagnosed by the 2 methods. The first one is forced swim test. Another test is sucrose consumption test. In this forced swim test, the experimental group rats were placed in the cylindrical container of diameter 10 centimeter, height which has a height of 25 centimeter, with a water level of 20 centimeter depth at 25 degrees Celsius for the total of 6 minutes. The initial 2 minutes were not considered the time period during which the animals tried to escape out, and which happened to be the final 4 minutes were considered to calculate the immobility of animals inside the water container. For the sucrose consumption test, after the induction of depression, the 1000 sucrose solution were given to the rats for 24 hours. After it has been absorbed for 24 hours. After the 24 hours, the consumption level will be calculated. After this confirmation test, the animals were, the group 3 and group 4 animals were treated with standard drug as well as the plant sample for 28 days. After the end of the treatment period, that is 28 days, the animals were sacrificed by cervical dislocation under mild chloroform anesthesia. Blood was collected by the cardiac sensors and the serum was separated by the centrifugation at 5000 rpm for 10 minutes. The brain was excised immediately and thoroughly washed with the saline for further use. After this, here I have given the result and discussion. The first result part is forced swim test. This is a confirmatory test for predict the status of depression with the time of immobility. The first swimming tests were taken at 4 times at different intervals. In forced swim test, albino rats are forced to swim in restricted space from which they cannot escape. This induces state of behavioral depression in the animals, which is claimed to reproduce the condition similar to the human depression. The result shows that Sesparia pipinosa extract can decreases the immobility time in forced swim test. After 28 days of treatment, it is found that the Sesparia pipinosa produce a higher anti-depression activity. This is similar to the standard drug imipramine 2. And in the sucrose consumption test, after the experimental period, that is after the 28 days, once again we have do the sucrose consumption test for confirming whether it has been in the depression state or not. So during that day, the group 2 shows the decreased level of sucrose consumption because the depression rat which won't consume more amount of depression. So in the present study, there was a significant decreases in the sucrose consumption of the depressed rat when compared to the normal rats. During the treatment, the significant value were found to be reversed. Group 4, that is plant treated groups, will be show the significant increases in the level of sucrose. And after the sacrification, the brain has been excised, which has been used for further analysis like enzymatic as well as the non-enzymatic. In enzymatic, the present study, there was a significant decreases in the SOD and catalase activity of depressed rat, that is group 2, when compared to the normal rat, that is group 1. During treatment, the significant values were found to be reversed. Group 4 were treated with plant extract of Sesparia pipinosa, where it shows the significant increases in the level of SOD and catalase in the brain of rats when compared to that of group 2, that is depressed rat. In rats treated with the standard drug, imipramine showed increased activity of SOD and catalase in the brain, which is close to the normal control. The decreased SOD activity in the depressed group may be because of highly reactive oxygen metabolite production. The decrease in the erythrocyte SOD levels were suggested to be a hallmark of depression. The antioxidant study in the brain tissue of depressed rats revealed decreases in the catalase level, denoting that effect of different triggered free radical generation. The experimental of glutathione peroxidation and the ascorbic acid that reveal the both glutathione peroxidase and vitamin C a significant decrease in group 2 when compared to that of group 1. In the plant treated groups such as group 4, the glutathione peroxidase and vitamin C activity was significantly increased in the group 2 rats. In group 3, the rats, the activity of glutathione peroxidase and ascorbic acid in the brain was increased more than the group 2. This study shows that the activity of glutathione peroxidase is reduced, which could be due to the exhausted adaptive response to the counter effect of increased oxidative stress. As well as the vitamin C is an anti-stress vitamin and may counter too much adrenaline. Vitamin C blocks the behavioral response to the dopamine and enhances the effect of neuroepileptic drug. Depression and anxiety is associated with psychiatric disorder, which are attenuated by adequate intake of vitamin C. So, from this study, the plant extract Sesparia pipinosa have a potent anti-depression activity. It has been concluded for my study. So after 28 days of treatment, it has been shows the improvement in the level of enzymatic antioxidant like SOD, catalase and glutathione peroxidase, as well as the level of non-enzymatic antioxidant vitamin C. Thank you. Hello, ma'am.
Yes, over to the judges.
Hello, Mary Joy.
Sir, hello, sir.
Yeah. So, have you analyzed the stress level of animal other than your forced swim test?
Sorry, sir. Can you repeat it, sir?
Apart from your forced swim test,
Yes, sir.
did you analyze any other parameter that evident the stress level of animals?
Sir, I have done two tests, sir. One is a forced swimming test, another one is sexual constitution test. But the in forced swim test, it has been taken for 4 intervals, sir. Like before treatment, I have treated the animals in the forced swimming test, and the 5th day, 9th day, as well as the
That I have seen from your results.
Yeah, okay, sir.
I think that is it enough to judge that animal is in stress?
We have some other test also, sir. But this is for my presentation purpose. I have given only 2 tests, but in my research purpose, I have added tail suspension also, sir.
All right.
Sorry, sir.
It's fine. It's fine. And this is still in the preliminary phase of your research. That do you think that a mere biochemical analysis of antioxidants would prove that your plant has antioxidant impacts?
Sir, I have done some more antioxidant too, sir. And also, now I am doing my molecular work also. I have taken 3 gene and identified the expression of that also. But it's an ongoing project. That's why I'm whatever I have finished till it, I have given for presentation, sir.
Because Mary, it's a suggestion that mere biochemical analysis, maybe it's important, but mere biochemical analysis cannot prove the scientific phenomena. As the scientific community is progressing more towards more results that are proven with more evidence and details, such as proof in are cross-confirming the same with protein level as well as gene level expression.
Yes, sir.
Like how do you substantiate that the biochemical results, mere biochemical results, can support your view, or can say your hypothesis is strong enough to prove scientifically?
Sir, for my project work, I had more enzyme and I have added more non-enzymatic as well as enzymatic antioxidant. But for this presentation, I given only 4. But already I have done many antioxidants as well as the carbohydrate lipid profile also I have done with this work, sir. But I did show that.
Did you measure ROS activity or ROS?
Sorry, sir.
Because did you measure ROS? You haven't measured ROS, like say in terms of liquid peroxidation or oxidative markers.
Yes, sir.
You have measured only antioxidant responses.
Yes, sir.
But I expected that what was the reaction for stress? How that stress was understood, other than forced swim test and sucrose consumption.
Uh, yes, sir.
I expected whether you measured uh, oxidative stress or ROS or lipid peroxidation. If you focus only on the antioxidant parameters.
Yes, sir. I have focused only on antioxidant parameters, but I'll include that also, sir. For
But without
further,
measuring the oxidative stress, you can't talk about response.
yes,
Am I
sir.
right? Am I right?
Yes, sir.
That's what I wanted to hear from you. When you talk about antioxidant response, you should have measured about the oxidative stress level.
Okay, sir. I'll include that also in my
Yeah,
further.
that would be better for your publication sake. As
Yeah,
well
okay, sir.
you can read all these genes and their expression pattern, as well as protein level expression pattern, so that can substantiate your results for a good publication.
Yeah, okay, sir.
Yeah. Thank you.
Okay. Thank you, sir.
And over to you, Doctor.
Okay, I thank both the judges for wonderful commentary session, and you have enlightened the students with your inputs. So now we will wind up the session, and tomorrow we will meet again. And we have four speakers for tomorrow, and we have the another poster session. So if judges would like to say a few words, they can just say about the work. I mean. Most of the abstracts we receive from our own institution, but other department, so biochemistry department. So if you have any inputs overall, may I know your opinion, both the judges?
Yes, Satya. The session was very informative because it was a very welcome diversion from my means normal plan-based work, and I gained very good amount of information session. I didn't expect that the session will be packed with very good content. Thank you so much for having me here, and thank you, sir.
Thank you. Thank you so much, Pratima.
Thank you,
It's
sir.
a pleasure to see you here.
I have to thank both of you for promptly accepting the invitation. So we will meet again tomorrow. Thank you, ma'am and sir. So we will.
Thank you so much. Thank you so much.
Take the document. Maybe within the evening, you can send me the document. That will be fine. So tomorrow we will be finalizing with the poster sessions, which is going to happen tomorrow, and we will announce the. Announce the first three best poster and best oral presentations at the end of the conference.
Sure. Thanks for the invitation, Dr. Satya.
You're welcome, sir. You're always welcome, ma'am. You're always welcome.
Thank you, Satya. I'll be sending the document maybe in half an hour.
Okay, ma'am. Thank you so much. Okay, guys. We'll wind up and we'll meet tomorrow. So by tomorrow, the session will start by 9:30. Have a good day, all.
Thank you, ma'am.